Thank you all of you for coming. It's a pleasure to be here. And we are delighted that so many people could join us for our virtual event. Our first speaker is Sarah Sankuhler, who is a MA, PhD candidate and a student in Dr. Samuel Mackenzie lab at University of Rochester in New York. Her thesis work focused on studies TANGO2 deficiency disorder in a C.elegans or worm model system. Sarah has worked very closely with Dr. Mackenzie on several of his foundation-funded research projects that are highlighted on the TANGO2 Research Foundation website. Welcome, Sarah Sankuhler. Thank you very much.
All right. All right. Thank you, Giorgio, and good morning, everybody. All right. Just working on getting my slides pulled up here.
Under the view options on the top, Sarah,
click request remote control.
That's it. And you'll have to click once and then it'll give you remote control.
All right. Let's see. All right. So I'm very excited to be kicking everything off for everybody this morning. And I'm really pleased to be telling you about the work that I'm going to be doing for my PhD thesis as a student, where I'm going to be investigating TANGO2 deficiency disorder in a C.elegans worm model. Right. Off the bat, I have no financial conflicts of interest to disclose. Wish I did, but that's not how graduate school works. All right. So just to give a brief overview for everybody, I'm sure many of you are familiar with this information, but TANGO2 deficiency disorder, which I'll refer to as TDD, is a rare genetic neurological condition. This is caused by biallelic mutations in the TANGO2 gene. So it's inherited in an autosomal recessive pattern. There are currently about 150 diagnosed patients worldwide. However, based on some studies of the frequency of alleles with this mutation in the general population, this condition likely affects about 6,000 individuals throughout the world. It's also important to note that TANGO2 is located in the 22q11 locus, which is the site of the deletion in DeGeorge syndrome, which is the most common microdeletion syndrome in the world. So these patients represent a significant population of people that are at heightened risk for having TDD. And then in this panel on the right, I just wanted to point out that there are a very large number of possible mutations or spelling changes within the TANGO2 gene that can lead to TDD. So most commonly, this can be a large deletion in exons 3 through 9 of the gene. So a very large chunk of the gene might be missing. But in other patients, you can also get spelling changes at a single nucleotide within the gene that can also cause disease in many of our kids with TANGO2. All right, so from a research standpoint, there are a lot of questions that we have about TDD that we don't have a lot of answers for. So we don't know what the protein TANGO2 really does in the cell with a lot of clarity, nor do we have a really clear idea of where within the cell this protein is working. And most importantly, there are still a lot of questions about why a lack of TANGO2 or mutations in the TANGO2 gene would lead to disease within some of our TANGO2 kids. So later today, you're going to hear some great talks from Dr. Sacher and Dr. Miyake about some of their findings. So I don't want to give the game away too much, but because some of what they found influences some of the questions that I have about my research, I did want to make mention of some of the things that they've noticed. So Dr. Sacher has some fruit flies that are missing TANGO2. And he has found that those fruit flies, when they are treated with vitamin B5, seem to have a protective effect against some of the symptoms of TANGO2 within these flies. So TANGO2 mutants that are treated with vitamin B5 seem to recover from the seizures that these flies can experience. And then similarly, Dr. Miyake, who led a natural history study of patients with TANGO2, has shown that patients that take B complex vitamins, which have vitamin B5 in them, don't experience metabolic crises, which, as many of you know, can be a really significant cause of complications and even mortality for patients with TANGO2. So understanding what vitamin B5 might be doing and how it might be helping patients with TANGO2 is a really big question. So just to go over a little bit of cell biology. So vitamin B5 is also known as pantothenate or pantothenic acid. And really the only known role that we know about for pantothenic acid in the cell is to be turned into a compound called coenzyme A through a synthesis process with a series of enzymatic steps that lead to the production of coenzyme A, which can then go throughout the cell and performs a really wide variety of functions. Coenzyme A is known to be involved in about 10% of all of the reactions that happen in a given cell at any time. So this is a really, really important molecule for cellular metabolism and cell health overall. So this is synthesized from vitamin B5 and then is, once coenzyme A has been used up, is salvaged through a complicated and not really well understood pathway so that it can be then recycled back into pantothenate and the process can continue. I also want to point out that since B5 seems to have some beneficial effect in TANGO2 patients or in Dr. Sacher's fruit flies, this would suggest that this pathway that allows you to synthesize coenzyme A from pantothenate still works, which to us suggests that if there is a problem with coenzyme A handling in TANGO2, that this problem would lie on sort of the salvage or degradation side of TANGO2 handling within the cell. So in order to ask some more questions about this and study this more closely, we need some good model systems that work really well. And so for my thesis project, I'm looking at making a model of TANGO2 deficiency in a nematode model, which are these tiny microscopic worms called C.elegans. So why is this a good model for TANGO2 deficiency disorder? Well, these worms are very, very well characterized. They've been studied since the early 1900s, and people have characterized their behavioral patterns very, very closely. So if worms are acting differently than we might expect, then that's something that we can be able to notice. These worms have a very short lifespan, so it's easy to study them from development all the way through their death, and they reproduce very quickly. So you can generate lots of worms in a very short amount of time and get lots of really good experiments done. These worms are hermaphroditic, so they produce eggs that they are able to fertilize themselves, which makes their genetics very easy to track throughout the different generations, and it's easy to manipulate these genetics as well. In order to reproduce, these worms have to generate many, many times their body weight in the eggs that they lay in a very, very short period of time, which means that their metabolism is highly active, and these metabolic pathways are very well conserved surprisingly enough, between worms and humans. So studying metabolism in these worms can give us a lot of insights about metabolism that goes on within humans as well. And then critically for us who are interested in TANGO2, worms have two known homologs to TANGO2. So there are two genes that are very, very similar to TANGO2. These are called HRG9 and HRG10. And as we can see on the bottom here, from a prediction of what the structure of these proteins would be expected to look like, here we have human TANGO2 is on the left, and HRG9 and HRG10 are to the right. And you can see here that the expected structure of these proteins is really quite similar and is quite well conserved, which may suggest that these proteins may be performing a similar function to what human TANGO2 does, which would make them really good candidates to study. To get a little bit more into what we think these proteins might be doing, which is a little bit of a hypothesis on my part, but I think could be interesting. So here we have the amino acid sequence of human TANGO2 on top and HRG9 and HRG10. And this is showing in these segments that have the dark background. These are showing areas where these amino acid sequences are really well conserved or are the same between TANGO2 and the C.elegans genes. So there are a number of places throughout these genes that have a lot of similarity between the two. And then interestingly, some predictions of what the TANGO2 gene might look like in these worms predict that it could form what's called a hydrolase fold. So this would be a potentially enzymatic site that could help break down proteins within the cell. And there are certain amino acids that would be important to the function of that fold that we've highlighted here in these sort of colorful segments. And interestingly, these segments that are part of this possible hydrolase fold are both really well conserved between human and worm TANGO2, and also are the site of some known point mutations or spelling changes within the TANGO2 genes that we know cause TANGO2 disorder within humans. So, which I've shown here in these red letters that are above the sequence alignment here. And so what this could point out is that if you have a mutation that disrupts this potential site, you could be disrupting the function of this protein, which is just something interesting that I wanted to point out. All right, so for my thesis work, I've been working on generating a model of TANGO2 deficiency disorder in the C.elegans worms using CRISPR technology. So this is a technology that allows us to go into the germline of these worms and make some genetic changes. Some of the data that I'm going to be showing you has been done in a separate line of worms that we obtained from a different lab that have a knockout of both HRG9 and HRG10. So these worms are missing both of these genes, but our lab has also been going through and making our own version of these. And so that's what I've been spending some of my time doing. So I just wanted to point that out, that we've been going in and making some genetic changes into these germlines and knocking out these genes within these worms. All right, so to get into some of my data. So the first big question is, do worms that are missing these two genes, which I will refer to from here on out as our double knockout worms, since they have knocked out two genes, do these double knockout worms have a phenotype or a behavioral profile that is different than our normal wild type worms, which in C.elegans, N2 refers to our normal standard wild type worms. And we do have a couple of areas in which these worms do have a different phenotype. So looking at how long these worms tend to live, our double knockout worms, as you can see from from some survival studies that we've done, our double knockout worms seem to have a reduced lifespan from the N2 wild type worms. There is an interesting caveat with some of the work that I've done here that I want to point out, which is that in doing these experiments, the worms live on these little on these dishes. And normally they tend to stay in the dish, which is where their food is, and they like to live. And so they'll stay in the dish and they'll crawl around until they eventually pass away. And then we can count whether they're alive or dead. However, our double knockout worms have a bit of a habit of crawling off of the dish and sort of escaping over the sides, which is contributing a little bit to some of the possible lack of longevity that we're seeing here, which there are a number of reasons why this could be happening, but this does tend to be a symptom of metabolic stress in these worms, which is why I wanted to point that out. We also, looking at some of their, at other key moments of their lifespan, looking at reproduction within these worms, normal wild type worms are known to lay broods of eggs of about 250 to 300 eggs. However, our double knockout worms lay significantly fewer eggs, so maybe about 100 eggs per worm, which is also particularly indicative of some possible metabolic dysfunction within these worms. Because as I said earlier, it's really metabolically taxing on these worms to generate lots and lots of larvae. And so the fact that these worms are laying significantly fewer broods are suggesting that these worms may not have the metabolic capacity to generate the expected number of eggs that one would think we would see. We also noticed through the course of studying our worms that our double knockout worms tend to eat a little bit less than our normal wild type worms. And so one proxy that is commonly used for this is that of pharyngeal motility. So these worms have these pharyngeal structures, which you can see here in this rounded shape that I will hopefully be able to play this video. Yes. So here's a normal wild type worm, and as it moves around, you can see its pharynx sort of fluctuating up and down within this circular structure in its head. And so this is a sort of known thing that you can sort of quantify and use as a proxy for how much food the worm might be eating. And so this is a little bit subtle in our double knockout worms, but through some careful scoring that was done by an undergrad in our lab, we can see that our double knockout worms have some pharyngeal pumping that tends to be about 20% slower than our normal wild type worms. So which there are also a number of reasons that could be contributing to this that are particularly interesting. But we also have some very preliminary data that might suggest that when we treat the food that we feed these worms with a small dose of vitamin B5, this might be able to restore our pumping rate back up to what we would expect in our normal wild type worms. So we are excited to see that there is possibly some benefit to vitamin B5 within these worms. All right, and then one more sort of bit of behavioral data that I want to go over is sort of an incidental finding that we saw, which is that our double knockout worms seem to have decreased motor function. So here we can see a view of the plates that our worms live on. And so they live in these lawns of E. coli, actually, that they crawl through, and you can see sort of the trails that they leave behind as they explore the plates. So these are views of a single worm on each plate, and you can see that the trails left behind by one normal wild type worm exploring the plate. These normal worms sort of crawl all over and go all around the plate, whereas these double knockout worms, you can see explore a lot less of the plate. So to kind of find a way to quantify some of the motor function within these worms, I developed this swimming assay. So swimming is a really metabolically taxing challenge for these worms. They have to be able to thrash their whole bodies, essentially for an extended period of time to be able to move through a liquid environment. And normal wild type worms should be able to maintain this thrashing for an extended period of time, up to about 90 minutes. And so in the course of this assay, I took some C.elegans worms, placed them in a buffer that is isotonic to them, so they won't have any negative effects from being within this liquid environment, and then scored them for their ability to swim throughout a 20-minute window. So if they were able to thrash rigorously throughout this time, they receive full marks. After a certain point, if they're still moving around but are unable to propel themselves through the liquid, they would then be scored a little bit lower. Let's see. And then I have some more videos to show you to illustrate what I'm talking about. All right, so that's playing okay. So this is an N2 wild type worm. So as you can see here, this worm is able to thrash its whole body in this sort of sinusoidal motion in a really vigorous fashion. He currently has a little bit of food that he's trying to eat, which is why he's kind of staying in one place. But you can see that he's able to sort of maintain this really vigorous motion for an extended period of time. On the other hand, we have our double knockout worms that are missing TANGO2. And so this worm has been in this liquid environment for the same amount of time as this wild type worm. However, you can see that it's not able to move nearly as rigorously as we would expect of a normal wild type worm. Which again would suggest to us that this worm may have some sort of metabolic inability to generate the energy needed to use its muscles to thrash really vigorously, like we would expect with our N2 worm. And then this is sort of a rough quantification of what I just showed you. So on the left, we have our normal healthy worms, and you can see that they're able to, throughout this 20-minute window, they're able to swim really vigorously for pretty much the whole time. One or two might take a break every now and then, but then they pick up swimming vigorously right back up. However, on the other hand, our worms that are missing our TANGO2 homologs on the left all the way on the right are our double knockouts. And then they sort of lose the ability to swim vigorously fairly quickly. And then interestingly, we also have some lines of worms that are missing only one of these genes. So just HRG9 or HRG10. And interestingly, they also don't have the ability to maintain this vigorous swimming for a long time, which is kind of an interesting question, because if you have two genes that have very similar structure and maybe similar functions, one might expect that they should be able to sort of cover for each other. So if you're missing one, the other one might be able to pick up the slack. But that's not what we see in this case, which we thought was very interesting. So then to kind of bring everything back to some of the data that I pointed out in the beginning, since we have seen maybe a little bit of sort of a bit of a teaser that vitamin B5 is able to help these kids, and vitamin B5 gets turned into coenzyme A within the cell, we were interested in knowing what's going on with the coenzyme A levels within these worms. So with the help of a collaborator of ours in West Virginia, Dr. Leonardi, we sent her some samples of our worms that she was able to look at the total level of coenzyme A within both our normal wild type worms and within our double knockouts. And so we sent her worms that had both been, that were fully fed and had been collected from plates where they had been fed, and also a sample of worms that were collected from plates after they had been starved for about 24 hours. And so our very preliminary results show that worms that are in the fed state seem to have similar levels of CoA pools throughout the worm tissue. However, worms that are lacking our TANGO2 homologs seem to be, when they're deprived of food, these CoA pools may be depleting faster within these worms, which is something pretty interesting, but would also support some of the beneficial effects that we're seeing from vitamin B5. And then our caveat here is that this was from just one sample. So we're sending her some more stuff very soon that we're hoping will further support some of what we've seen here. All right, so just to kind of wrap everything up, I want to talk about the hypothesis that I'm going to be testing within my thesis. And then just to point this out, this is just sort of our hypothesis. So this is sort of the framework that I'm approaching some of the questions that I want to answer during the rest of my years of my PhD. So this is what could be going on, but I want to see if this is true or not. So we have this, so in your cell, you have this pool of coenzyme A that's really important for a bunch of different metabolic pathways. And this pool of coenzyme A is supported through both synthesis, which is where vitamin B5 is able to play a role, as well as salvage of CoA, which may or may not be where TANGO2 is able to play a role. So what might be going on is that if your cells don't have TANGO2 or don't have its homologs like AGG9 and AGG10, this salvage pathway may not work properly. And in various tissue types, this synthesis pathway may not be able to adequately cover for to restore this pool of coenzyme A. So coenzyme A is less available to be used throughout the cell for the very, very wide variety of functions that it needs to be used in. However, then when more external vitamin B5, through in TANGO2 kids, through maybe the use of some supplements, is delivered into the cells, this might be able to drive forward this de novo synthesis pathway a little bit further. That might be just enough to restore this pool of coenzyme A to the amount that needs to be available to then be used throughout the cell again, and restoring the cell to a metabolic state that is more beneficial. All right, and then, so then for the rest of my thesis, I have a lot of questions that I would like to answer. I have plans to continue to fully characterize the behavioral phenotypes that I've seen in my worms, and really dig into what stress can do to these worms in terms of their behavior and also in terms of the CoA profile within these worms. And find out if the behavioral phenotype I'm seeing really truly correlates with the coenzyme A levels within these worms, and whether treatment with vitamin B5 is able to restore either the CoA levels or the phenotype that we're seeing. And then also dig further into the role of TANGO2 in different tissue types throughout development. All right, and with that, I just want to thank everybody who has helped me with my project along the way. Sam's been an awesome mentor who never fails to provide donuts, as well as other kinds of support, of course. And also our lab tech, Kayla, who has been invaluable in helping me with all of our experiments and keeping things shipshape. The Sun Lab for giving us the worms that some of this data was generated on, as well as the TANGO2 Research Foundation for funding us and supporting a lot of our work. And with that, I would be happy to address any questions.
Hello, I'm Jamie, mom to Oliver, who has TANGO2 Deficiency Disorder. I am pleased to introduce our next presenter, Dr. Agustin Lujan. Dr. Lujan is joining us today from Barcelona, Spain. Dr. Lujan is a member of Professor Vivek Malhotra's lab at the Center for Genomic Regulation in Barcelona. His research suggests that TANGO2 is involved in acyl-CoA metabolism, thereby controlling the cellular levels of various lipids and mitochondria functions. Dr. Lujan is passionate about science, technology, and education, particularly related to new health technologies and triple impact projects. Dr. Lujan.
Okay. Hello, everyone. Can you hear me? Yes. Okay. So, well, first of all, thanks the organizer for giving me the opportunity to present our data on the functional significance of TANGO2 in the cell physiology. And thank you, Jamie, for your introduction. I want to start this presentation, if I could have the control. Yes, it's a little bit. Okay. So, well, first of all, I have to say that I have no financial disclosure or conflicts of interest with the presented material in this presentation. And I want to start the introduction talking about the secretory pathway. As many of you know, the Malhotra lab has a long history studying the secretory pathway. And this pathway, it's by which cells produce proteins, modify these proteins, transport, and also secrete it outside or inside the cell. And this is very important because these proteins are essential for the homeostasis of the cell. So our lab, it's a lab specialized in the study of this secretory pathway. So in 2006, in the Malhotra lab was performed an extensive screening that revealed that different genes, when you delete it from Drosophila cells, Drosophila, it's a fly, when you delete those genes, result in some kind of defects in the secretion or the organization, the structure of the Golgi. And that's why we call TANGO proteins, because it's the acronym of Transport and Golgi Organization Proteins. So in the last 15 years, more or less, the lab has extensively worked on the function of TANGO1. And this protein is essential in the collagen transport. So the lab described and demonstrate that TANGO1 has a very important role in the collagen export. And this was something that was studied for the last 40 years. And our point of view solved a huge problem in the field. But today, we're here because we want to talk about TANGO2. So 2 years ago, when I joined the lab, I began to analyze these other genes from the TANGO screening. And then Googling, I found a human disease that was related to TANGO2 genes. And looking at the TANGO2 Foundation research, I found a lot of information that was very important for me when I start this project. So for all these reasons, I decided to deepen our research on this gene. So today we are talking about this TANGO2 gene in humans. So let's go to what we know now about TANGO2 gene. So the first peculiar observation was that TANGO2 is highly conserved in very different species. And this is not something normal in all kinds of genes. So we can find TANGO2 from plants to humans, in yeast, in flies, in fish, in amphibians. So that means or suggests us that the function of TANGO2 has to be basic throughout the whole evolution. So then, looking at what was published about TANGO2, we were pleasantly surprised to learn the work of our college at the TANGO2 Research Foundation, and looking the clinical features and the medical characteristics that appears in this TANGO2 deficiency disorder caught my attention the evolution of this metabolic crisis. Because if we have to talk about metabolic crisis, it's important to understand that the human body uses carbohydrates as the primary source of quick energy. So when we eat more calories or carbohydrates than what we need, we store that calories as lipids. So we are putting our energy in terms of lipids to store them for the moment that we need more energy. What I want to point this out, because the metabolic crisis in TANGO2 patients appears after two different, or mainly two different periods. One is starvation and the other is infection. In those situations, the body needs a more amount of energy. When we are under starvation, for example, we use the energy that is provided from lipids to produce ATP. So we consume our carbohydrates and the body uses lipids to produce energy. We move these lipids that are packed in structures that are called lipid droplets, like droplets of fat. We move those lipids to the mitochondria, and the mitochondria produce ATP. It's like our factories in which we want to produce something, so we put a source, we produce our energy, and the factory also produces CO2. In case of our cells, this CO2 that it's producing, it's called ROS. ROS are the reactive oxygen species. So mitochondria produce ATP, that is our kind of energy in the cells, using lipids, but also producing something that we don't want too much in the cell, that is ROS. So taking this as an important message, we start to think, okay, what's the function of this TANGO2 protein that when you don't have carbohydrates in your blood, or you are in starvation or infection because you need more energy, then appears this metabolic crisis. So the first thing that it's important to understand when we are studying a new protein, a new gene, is to localize this protein in the cell. Because when you localize a protein in the cell, then you can understand more or less which pathway, which physiological pathway it's working on this protein. So you could have a probable function of your protein. So first of all, what we did was a straightforward experiment in which we put a tag on TANGO2. As you can see here, we put a GFP tag that it's protein fluorescent in the green channel of the microscope. And also we did it with M-scarlet, that it's a tag protein in the red color. So in confocal microscopy, when you have two proteins at the same time in the same place, you can see in yellow in the merge, like here. So now that we had this protein with the tag, we can analyze the location in the human cells. So we use different markers, well-known markers for different structures in the cells. And as you can see in the top part of the figure, we put TANGO2 in the red color, this one, and a mitochondrial marker in the green color, that is this one. So when you see the merge, as we found a lot of yellow in this picture, seems that TANGO2, the isoform 1, and mitochondria are in the same location at the same time in our system. So TANGO2 is mainly at the mitochondria in this sample. But also, we check other kind of compartments in the cells, like the ER, that it's a compartment in which we produce most of our proteins and also lipids. And as you can see here, we have some dots or some areas of colocalization between our TANGO2, red here, and the ER marker. So it's not the same as the mitochondria. There is almost a perfect colocalization, but we have some sites with the colocalization between TANGO2 and the ER. But this doesn't happen with all the markers that we use. For example, here we use a marker for peroxisomes, that is in green. And as you can see here, there is no colocalization between TANGO2 and the peroxisomes. But when we use a marker of lipid droplets, these structures that I told you that we use as a storage to store lipids, we saw again some points of colocalization between TANGO2 and the lipid droplets. So to be sure that we were looking real contact sites between ER and TANGO2 and lipid droplets and TANGO2, we analyzed TANGO2 and these markers in different point of view of the cells. So we analyzed in orthogonal views with in XY, Y zeta, and X zeta, and we found the colocalization in all of the planes that we analyzed these contact sites. So, and now we can say that TANGO2 is predominantly at the mitochondria. There is a small pool in the cytosol also, but predominantly it's on the mitochondria. And there are some colocalizations between TANGO2 and lipid droplets and also the ER. So a second option to study a new protein is to remove it from the system. So if we have a cell and we remove a protein, we can see which kind of phenotype or which kind of balance the cell produce to survive without this protein. And this is what we did after the localization. We removed TANGO2 in a transient mode in which we silenced the gene for 48 to 72 hours. And what we found was that the lipid composition changed. As you can see here, we use a marker of lipid droplets. It's a marker in which when you put it in the cells, all the neutral lipids are labeled by a red color. So as you can see here, in the control cells, in blue, you can see a DNA marker, so the nucleus of the cell. And in red, you can see these lipid droplets. This is the control. And this is the size of a lipid droplet in control cells, in hepatocyte cells that we are using in this experiment. But if we remove TANGO2, if we deplete TANGO2, what we can see is that the lipid droplets are higher. So the size of this lipid droplet is bigger than in the control situation. So we developed a program to put all our pictures or our images of the confocal microscopy and to analyze them without the bias of our selection. So after the analysis of all the pictures that we took in the microscopy and in the confocal microscopy, what we revealed was that the total volume of lipid droplets was higher in those cells that doesn't have TANGO2. But also the volume of each lipid droplet was bigger than in control situation. But in terms of the amount, the number of lipid droplets was the same. So in an acute silencing of TANGO2, what we saw is that we had lipid droplets that are bigger than in control situations. So now we know that TANGO2 is at the mitochondria, contact lipid droplets, contact the ER, but when we remove the gene, we have big lipid droplets. So there is two possibilities or three here. If we have a lipid droplet that it's like a bowl, if we have a bigger lipid droplet, it's because we are putting more lipids inside, or it's because we are blocking the exit of these lipids. This is the two possibilities that we have to have a bigger lipid droplet. Or both. This is the third option. So, as I don't have too much time to show the experiment, but I have extra slides if you want to see it in the QA. We make two different experiments in which we block the biogenesis, so the production of new lipids, and also we block the lipolysis, the exit of these lipids to be used by the mitochondria. And what we saw was that in those cells that doesn't have TANGO2, there is an increase in the biogenesis, so there is an increase in the production of lipids, but also, and it's even bigger, there is a decrease in the lipolysis, in the catabolism of these lipids. So the cells, it's unable to use these lipids to produce more energy. So now we know that we have more lipids inside the lipid droplets, but what kind of lipids are affected in the TANGO2 depleted cells? Because now we have an idea that there is a problem of the quantity of lipids, but we don't know if it is also a problem in the quality or the kind of lipids that we have in our cells. So next idea was to study what kind of lipids are affected in TANGO2 depleted cells. And to remind you, this is one of the most common graphics that you can find to explain the metabolism of lipids. And as you can see here, this compound here called PA, or phosphatidic acid, it's a key compound in the metabolism of lipids, because from PA you can produce different kind of phospholipids, or you can store them as deacylglyceride and triacylglycerides in these lipid droplets. So we send our samples to Germany to study this kind of lipids using lipidomics. So in our system, what we did was to prepare 4 different samples. First one, we use control cells in nutrient conditions with all the nutrients, amino acids, carbohydrates. Second sample was the control cells, but in starvation, under 4 hours of starvation without glucose. So those cells needs to remove lipids to produce ATP. The third sample was cells with all the nutrients, but without the TANGO2. And the fourth sample was cells without the TANGO2, but under starvation. So as you can see here, in normal situation, in a physiological starvation, cells use LPA to produce PA. So in red, it's less. In green is more of this lipid. So if we did a ratio between starvation and control conditions, we can see that in a physiological starvation, we are reducing this compound here, LPA, to produce this compound here, PA. Because from here, we can go everywhere in our lipid metabolism. But when we delete TANGO2, what we found is that we have an increase in LPA and a decrease in PA. So we are having an increase here and now a decrease here. And how it is possible? So maybe there is something wrong here in the middle. And when we start the cells, was even higher the increasing in LPA, and also there is a decrease in PA. So we start to think that maybe TANGO2 is working at this level, in this stage, in which you can use all PA to produce PA, lysophosphatidic acid to produce phosphatidic acid. But how we can move from LPA to PA? Well, there is an enzyme here in the middle that are acyl-transferases. So to produce LPA, you need to take glycerol 3-sulfate, remove one proton, put an acyl chain, and this is done by an acyl-transferase called GPAT. And to move from LPA to PA, you need a second acyl-transferase that are called AGPAT that remove another proton and put another acyl chain. So if we think that TANGO2 is working here, we have to study those enzymes also. So this is what we did. We look the expression of these enzymes in our system. And what we found was that when we remove TANGO2, the cell balanced the absence of TANGO2, producing more AGPAT, so producing more this enzyme here, this acyl-transferase here, trying to move the LPA to produce more PA. But when we starve the cells, what we see is that the cell not only produces more AGPAT1, also AGPAT2 and AGPAT3. So most of the isoforms of this acyl-transferase were activating by the cells trying to producing PA from the LPA. So that could be explained by three possibilities that we are thinking nowadays. First of all, TANGO2 could be an acyl-transferase per se. Second, TANGO2 could be an acyl-CoA binding protein that moves acyl-CoA from one side to the other to give it to these acyl-transferases, or also could be a coenzyme of these acyl-transferases working together with these acyl-transferases to produce this movement. So now we are collaborating with a group in Japan to solve some of our ideas, and we have a lot of new data to be published soon. And finally, as we saw that there were some published data about that when you don't have TANGO2, you increase the production of ROS, this reactive oxygen species that were produced by the mitochondria. We want to know if in our system this ROS was also increased. So we test the ROS species in our hepatocytes that were deleted from TANGO2, and we found that there is an increase in the production of ROS in those cells that doesn't have TANGO2, and it's even bigger when the cells are under starvation. But also, and I want to thank a lot our colleagues in Baylor and the TANGO2 Foundation, and also patients and family that provide us fibroblasts from patients, because was very clear that when we measure the ROS production under starvation in fibroblasts from patients was an increased production compared to the control. So this is very important for us to validate our model with the human fibroblasts. So now we know that in our system, it's also increased the amount of ROS that is produced in those cells that doesn't have TANGO2. But this increase in ROS is enough to modify our cells, it's enough to modify our lipids. So after that, what we looked was if our lipids were modified by lipid peroxidation. So we use a marker that when there is an oxidant in the media, change the color from the red to the green. And we can measure this ratio in our cells. So as you can see here, this decrease is the decrease in the ratio of reduced lipids compared to oxidized lipids. What it means is that there is a high lipid peroxidation of our lipids. So now we know that TANGO2, it's mainly at the mitochondria. It's contacting lipid droplets and ER. When we don't have TANGO2, we have big lipid droplets. This big lipid droplet has particular lipids inside. And also now we know that most of our lipids in cells that doesn't have TANGO2 are peroxidated. And what does it mean? Does it mean that maybe the lipids that are inside our lipid droplets are oxidized? So we cannot use these lipids to produce ATP. We cannot move lipid after peroxidation to the mitochondria to produce ATP. So finally, I want to thank you, Vivek, for his support. I want to thank all my colleagues in the lab that work and collaborate with this project. I want to thank our collaborators in Baylor because it was very important, our meetings with CIMA and also to receive the fibroblasts from patients. I want to thank also our facilities and other collaborators here in Spain and in Japan. Our grants, EMBO and Marie Curie for my fellowships, TANGO2 Research Foundation for their amazing work and to take care about patients, but also families. And if someone is interested in our published data, you can scan this QR. And thank you, everybody, for your attention. And I'm happy to receive your questions. Thank you.
I'm David Longman from Australia. I'm a TANGO2 dad as well as a member of the TANGO2 Research Foundation Outreach and Research Committees. I'm honoured to introduce our next presenter, Dr. Joshua Meisner. Dr. Meisner is an early-stage clinical scientist in paediatric cardiology at the University of Michigan Congenital Heart Center. He specialises in cardiovascular genetics with multidisciplinary collaborative clinics in RASopathies, cardiomyopathies, connective tissue diseases, and neuromuscular disorders. His translational science laboratory focuses on the mechanisms of MYBPC3, related hypertrophic cardiomyopathy, and TANGO2 deficiency disorder, utilising induced pluripotent stem cell cardiomyocytes and novel mouse models. He loves the TANGO2 families he has cared for and supports the foundation through his work on the research committee in addition to his research. Welcome, Dr. Meisner.
Thank you, David, for that nice introduction. And I'm excited to talk to you all about our work modeling TANGO2 deficiency disorder through iPSC cardiomyocytes. So I have no disclosures or conflicts of interest that I'd like to talk about. There we go. And as has been nicely explained by our last two presenters, TANGO2 deficiency disorder is caused by loss of function in both alleles of TANGO2, has multi-organ system involvement with these intermittent metabolic crises. And of these crises, the highest mortality is related to the cardiac involvement. And we have these multi-organ system, we have this multi-organ system involvement from the neurological component with development delay, ataxia, the transient TANGO2 spells, the metabolic crises with hypoglycemia, altered fatty acid oxidation, rhabdomyolysis, and then the cardiac involvement. And of these systems, one of the more quantifiable changes and reproducible changes during metabolic crises tends to be the cardiac phenotypes with a very clear change in terms of quantitative shift in QT prolongation, as well as ventricular arrhythmias and transient cardiomyopathy. And as we've discussed in the last two presentations, the mechanism is still unknown, although we're seeing clear evidence of kind of an expanding understanding of the potential sources of the mechanisms of TANGO2 deficiency, with a lot of stuff centering around mitochondrial function and how these cells process energy. So there's evidence for intracellular transport dysfunction, secondary mitochondrial dysfunction, the role of potentially heme, although that's becoming a little less so, and evidence of B vitamins kind of throughout this process. But one of the features that has been known, and we've kind of talked about during the last session, is that within the first clinical cases that have been reported, is that there's this shift, these molecular signatures of kind of altered fatty acid oxidation. So how does the cell process fats to demand energy? And the prominent phenotypes that we see from skeletal muscle, cardiac muscle, are cell types that are highly dependent on using fat for energy handling and kind of the increased energy production that comes from processing long chain fatty acids. So our work focusing on TANGO2 deficiency is using induced pluripotent stem cells. So that's taking a skin sample or potentially blood sample. We use skin in our lab, growing out the cells that come from the skin, reprogramming them back into pluripotent stem cells, which can be expanded and can differentiate into any type of cell type, any type of cell tissue type in the body. In our case, we look at cardiomyocytes. And this provides some important advantages. So as we've talked about, there's advantages for doing different animal models that we can see from C.elegans, the work in zebrafish, the work in flies, all have a lot of important information, but they're not exactly human physiology. And there are some differences. There's uniform and kind of fundamental mechanisms that you'll be discovering in all of the different models, but there can be some different translational differences between taking findings in other animal models and putting them in humans. So it allows us to replicate actually human physiology and human cells, but being able to do this in a lab with kind of the flexibility that you get from in vitro assays and being able to adjust different tissue types over time. And allows us to look for, particularly with reprogramming of cells, to look for the different clinical variabilities within individual patients that we do see in our family members. One family member is affected a little bit differently. Each of our patients are affected a little bit differently. And so you can look for some of the variability that we see in disease expression within these different cell lines. And you can do genetic manipulations in these and within one cell line to show how a lot of different variants can impact the TANGO2 function and potentially disease outcomes. So the next question is, why cardiomyocytes, which as they're one of the key causes of mortality related to TANGO2 deficiency disorder, although ideally with the kind of improving or kind of increasing use of B vitamins, hopefully we'll be having less cardiac crises and less metabolic crises, which is an important outcome from the work we've garnered so far through the TANGO2 Research Foundation. But it's a very clinically important model with very kind of direct outcomes from looking at these cardiomyocytes. And as I said, one of the more reproducible findings is during metabolic crises is QT prolongation and kind of the electrophysiological phenotype of how these cells are using electricity to pace and beat regularly and how they're able to repolarize that electrical activity. And the fact that cardiomyocytes, again, are highly dependent on oxidative phosphorylation of fatty acids, which kind of all points to these cells should be particularly sensitive to loss of TANGO2 function, and ideally should serve as a good model to give us the best signal to noise ratio for helping to tease apart molecular mechanisms and potential therapeutic options to be able to help start that translation from preclinical testing towards human testing. And so that was the goal of our kind of work that we proposed to the TANGO2 Foundation, was to do a systematic test of metabolic conditions within our TANGO2 deficient cell lines and control cell lines and do a detailed assessment of the electrical and mechanical phenotypes with the goal of doing secondary testing once we have a very established signal to kind of start to translate and potentially test available therapies like B5, like other kind of adjustments to mitochondrial metabolism and potentially gene replacement through AAV mechanisms. But we first need to establish a really good system that shows us clear signal to noise ratio in a clinically relevant model. But the advantages of iPSC cardiomyocytes do not come without the downsides. Every system has certain advantages, every system has certain limitations. And some of the challenges of iPSC cardiomyocytes is that within individual cell lines, just as in patients, there can be a wide range of variability. So it's really important when you're assessing your response and how loss of TANGO2 is affecting function, is to do that ideally within an individual cell line, so you can remove kind of the variation that you have when you collect cells from one donor, translate it or reprogram into iPSC cells, then doing it within multiple donors and multiple different patients. So it improves a conversion, again, kind of signal to noise ratio. In addition to that, there's a lot of variability between taking iPSCs, so these stem cells that can become any cell in the body, pushing them using a special combination or combinations to push them to become cardiomyocytes that are initially very immature, much more fetal-like and embryonic-like, to more kind of early stage, kind of neonatal, but they're still immature cardiomyocytes, but trying to push them as far along as we can. But as you go through those individual batches, there can be some variability. So ideally, you would test within a given batch to, again, help improve signal-to-noise ratio. And as you kind of go along from stem cells to immature to a little bit more mature cardiomyocytes, but still remain somewhat immature, we can get this change in both contractile and particularly important related to TANGO2 deficiency disorder is kind of the metabolic function, going from much more glycolysis-based to fatty acid oxidation-based. And many models related to iPSC cardiomyocytes are done in these 2D monolayers. So when we're looking at contractile and electrical function, there is some limitations in being able to quantify the contractile mechanics. So as you can see here in just live cell imaging of cardiomyocytes beating, they're exhibiting regular beating, but they're often in monolayers in these disorganized networks where cells are pulling in different ways, and be able to quantify by looking at this active stress fibers in the same bright field image over on the right-hand side. You can see how individual cells are moving in different directions, and quantifying these mechanical changes can be difficult to be able to really assess how loss of TANGO2 and how different metabolic stress conditions are changing the mechanical properties and the contractility within these cardiomyocytes. So to help with that, we developed a system that allows for kind of using spatial cues and kind of micro pattern techniques to allow these cardiomyocytes to form in much more kind of organized structures that you would see it within the heart itself, but within organs themselves that are aligned uniaxially. So contraction be coordinated. So in a bright field image, just non-invasively looking at these individual cardiomyocyte muscle bundles, you can see they contract in the same direction, going from ends towards the middle. You get these very nice, replicatable, and kind of quantifiable changes in terms of contraction. But you can do this on a scale where you're able to kind of assess, and you're able to assess tens to hundreds of individual tissues within a given batch, within a given cell line, to give us really high kind of precision within each individual experiment and each individual testing condition. And so, again, kind of looking at these contractile mechanisms, just doing non-invasive bright field imaging, we're able to kind of look at displacement of these contractions to measuring kind of a shortening fraction, which kind of gives us this measure of contractility. Looking at the contractile velocities, both in terms of how it's contracting, how it's relaxing, to really understand the mechanical properties that are happening or changing with different stress conditions and with and without the presence of TANGO2. And as we're able to rhythmically measure and kind of quantify contractile assessment, it also gives these surrogate markers of electrophysiology in terms of the duration of contraction, which is proportional to action potential duration, as is a measure of QT prolongation, the rhythmicity of the contraction, how regular it is, are there kind of early kind of intermittent beats that you'd expect to see, kind of the surrogate marker of what you would see with electrophysiology that are associated with ventricular arrhythmias, which are, again, kind of the primary mechanisms of mortality and TANGO2 deficiency disorder. So kind of looking at our process of taking stem cells to cardiomyocytes from their immature state to the more mature state, but kind of more heterogeneous and monolayer to our two-dimensional muscle bundles. It's a relatively long process with a lot of different conditions you can vary. So we started as our iPSCs using Wnt inhibition, followed by Wnt activation, using lactate purification, and then trying to promote, able to kind of push them towards immature cardiomyocytes, do selection to select out non-cardiomyocyte cells, and able to save them down for later studies. Are we able to take them on to the monolayers, where we help develop and push them further towards increasing maturation, increasing use of metabolic and oxidative phosphorylation. So the higher metabolism, how those cells handle fats, and kind of the more advanced, more adult-like physiology, which we're able to push further by using our muscle bundle preparation. And together, using this combination, we're able to achieve like we're able to achieve more precision and be able to assess kind of a tighter assessment of contractile properties and functional properties compared to traditional monolayer assays. So this being a inhibitor of myosin, which is, it decreases the ability for those cardiomyocytes to contract. Whereas in monolayers, we're doing the same dose, they're being affected in the same way, but because of how you're able to assess those contractile properties and how they've matured in this more heterogeneous organization, we really do not, you can see there's small changes, there are significant changes, but they're very small, and being able to use these changes to really assess molecular mechanisms and potential therapeutic options is going to be very difficult. Versus our two-dimensional muscle bundles should give us more power to assess these changes within individual cell lines, within individual batches and testing conditions. The muscle bundles also give us a little kind of more adult, more mature electrophysiology properties in terms of ion channels, which again should give us the best chance to be able to assess the signal in the noise that is in vitro assays. So to be able to assess how loss of TANGO2 could be affecting how these cardiomyocytes are able to function in terms of their electrical activity, in terms of their contractile properties, we chose a couple of different methods for testing. So we can take our control cell lines, again, make them, kind of push them into stem cells, and then to help isolate the effects within an individual cell line, we're able to do CRISPR editing to knock out TANGO2. So that is our gene-edited cells here, the T2GE compared to our control cell line. So this is within an individual cell line, should be the tightest correlation between the two, as well as having patients. This we were able to compare to additional control cell lines. So once we're able to establish a very clear difference between isogenic, so within a same cell line differences, then we should be able to move on to kind of more expanded phenotyping, confirming that in additional cell lines. And so these are just examples of muscle bundles created from our control cell line and our gene-edited knockout cell line for TANGO2. They're able to form nice, clear muscle bundles, have regular rhythmic contractions that we're able to assess their function. So this gives us and sets us up to do an extensive battery of metabolic testing. And as with all science, there is a wide range of conditions almost an infinite number of combinations you can test. So you have to choose this in a very organized method. And as related to our testing system here, we're able to adjust kind of the maturation base to see how the effects of having kind of baseline higher amounts of calcium closer to physiologic levels that promote contractile maturation. So the cells are actually contracting stronger with higher calcium. But coming with higher iron levels, higher B5 levels, which are not higher, but they're just closer to physiologic levels, versus lower calcium with low iron, low B5 levels, and then be able to control what are their energy sources available to mimic both maturation in terms of how they handle energy, and in terms of their different stress states. So with or without galactose, with or without glutamine, with or without glucose, lactose, pyruvate, with or without lactate, with or without long-chain fatty acids. So our methods have been, we replate into the two-dimensional muscle bundles, allow them mature for 7 days under our different maturation conditions, and then perform stress testing with different metabolic stresses, whether that's with glutamine only, with lactate or pyruvate only, with fatty acids only, with or without heme stress stimulus, in multiple different cell lines, with our primary focus being our isogenic controls versus our TANGO2 gene-edited line, and within interesting findings, we're replicating that in our control and patient-derived lines. So all of these combinations produces a massive amount of data that I will not be able to go through. So I'm just going to focus on one example and show you the type of data that we look at and kind of get to our fundamental conclusion. We're looking at our control cell lines, which are here in red, versus gene-edited cell lines in blue. So this is an example of our testing conditions with low iron, low calcium, low B5, our PMI, B27 plus, or kind of supplement, using energy sources of glutamine and galactose as basal conditions with fatty acids, and then removing galactose, which is able to go through glycolysis, and lowering the amount of glutamine along with keeping fatty acids for 24 hours. We can see their baseline frequency with metabolic stress, so kind of the open circles, which are baseline conditions before, and then after metabolic stress, we're able to go back to the same exact tissues within the same exact batch, within the same exact cells, should give us the highest ability to assess differences related to metabolic stress, that there's no changes within our control cells. There's no changes within our TANGO2 gene-edited cells. There's a slight increase in terms of baseline frequency of the gene-edited cells, but within individual replicates, these replicates themselves had a slight difference, but there is some variation in that, so that there's kind of across a lot of different conditions, there's very minimal differences in terms of beating frequency. In terms of the rhythmicity and kind of the regularity of the beating, there's no differences between our control cell lines, between gene-edited cell lines, with or without metabolic stress. The overall contractile duration, they speed up during metabolic stress in kind of multiple different metabolic stresses, but they speed up to a similar degree and kind of can shorten their contractile duration to a similar degree. It's kind of the opposite of QT prolongation in both the wild type and gene-edited cell lines. And then we're going to go back. In terms of the contractile properties, we show these similar differences where the overall contractile strength, so kind of the fractional shortening, shows very little differences between our control cell lines and gene-edited cell lines. This particular condition shows a slight difference. But again, even replicating this, with the degree of replicates and the change, we're able to show significance with just a huge numbers of kind of comparison of muscle bundles. But this is not going to give us a signal to noise ratio to be able to assess and kind of probe the kind of fundamental molecular mechanisms and potential therapeutic options. And when you normalize this to kind of the basal max fractional shortening, the actual like fundamental contractile mechanics, there is no difference between there's no significant differences between kind of baseline conditions of control versus gene-edited cell lines, both in terms of contraction and relaxation. So we're using this very powerful model, and we're not showing very prominent contractile or electrical phenotyping at baseline across kind of multiple maturation conditions, multiple batches, multiple cell lines. And so there's, even with the small differences that we do see under certain conditions, there's not a lot of signal to be able to test for kind of differential effects of therapeutics. And it kind of shows the challenges of being able to kind of demonstrate a very clear phenotype in TANGO2. And I guess that kind of our next steps are to try to assess try to expand our thought process, expand the potential test conditions for metabolic stress. So while we've been reducing basal energy sources, we should be able to increase the stress, which we would kind of expect to see during the actual metabolic decompensations in patients, often associated with illness in addition to fasting. So they're burning through calories a lot faster when kids are sick, and they're eating less. We're going to try and replicate those same sort of conditions by causing these cardiomyocytes to beat harder, so giving them a little contractile agonist in addition to reducing their energy supply to really increase that metabolic demand and hopefully pull out some big differences. And we've been primarily using kind of these surrogate markers for electrical phenotyping. We'll move to more direct kind of electrical assessment using voltage sensitive dyes to be able to directly measure action potential kind of rate and rise time of these cardiomyocytes. So our ultimate goal is to kind of demonstrate a very clear cardiac phenotype, which I think we have the best option in our human cell lines based on kind of what we see clinically. And to be able to do some systematic testing of potential therapeutics, and again, kind of ideally kind of moving towards gene therapy as well, depending on what kind of how we're able to kind of show the potential therapeutic options. So with that, I would like to acknowledge kind of our lab members, our funding sources, in particular, like the TANGO2 Research Foundation has been really critical for promoting all the advancements we're hearing today and all the advancements we've heard over the last several years. The TANGO2 Research Foundation has really been important for coordinating those efforts, from the natural history study to the work in flies and kind of just the discovery of the utility of B5 and kind of how that's impacting patient care.
Hi, everyone. I am Debbie DeLoach, and I am the angel parent of Robert, who was affected by TANGO2 Deficiency Disorder and 22q11.2 deletion syndrome. I am also an active TANGO2 Research Foundation Early Detection and Diagnostic Committee team member. Our next presenter is Dr. Christina Miyake from Baylor College of Medicine in Houston, Texas. Dr. Miyake is an associate professor in the Department of Pediatrics at Texas Children's Hospital, where she serves as the director of the Cardiovascular Genetics Inherited Arrhythmias Clinic. In addition, she is an associate professor in the Department of Molecular Physiology and Biophysics at Baylor College of Medicine. Dr. Miyake is one of the investigators who first discovered TANGO2 deficiency disorder in 2016. I am now happy to turn it over to Dr. Miyake.
Hi, thank you, everybody, and thank you, Debbie. I didn't realize you were introducing me. So it's really a pleasure to be able to give you an update today on the natural history study. And I'm going to talk a little bit about the clinical relevance of the things that we've discovered through this study. And today I want to highlight how just individual families and patients can truly make a difference to the whole community. All right, so this is just a brief update. For the full information, it's quite a bit. I do plan to present that at the upcoming family conference in June of 2024. But just going back, many of you know that TANGO2 deficiency disorder, I'm going to call it TDD or just TANGO2 for short, was discovered in 2016, and Dr. Leilani really led all the efforts and really deserves the credit for this discovery. So after its discovery in 2016, really the question was, what is TDD? And we focused on the patients first to really guide our understanding. And again, I want to give credit to really those forces that made this study work, and that's through the TANGO2 Foundation, the conferences, Dr. Leilani, and the Morris family, who really have made the true difference in pushing this forward. So back in 2019, our natural history study began, and we started to collect data from around the world. If you look at what's happened over the years, in 2019, we had 22 children from 18 families, and that has increased over the years. We just had our 100th child enrolled this week, and we currently have 88 families. And that represents participants from 18 different countries. So in summary, we published our data in 2023, and that data encompassed information between February 2019 to May 2022. There were 73 children and now young adults involved from 57 families, and the ages range actually from fetal to 31 years was our oldest, with the median age of the participant being 9. About 50%, you've already heard, had the most common exon 3 to 9 deletion. 65% of the participants had suffered a metabolic crisis in their lifetime. 65% of them had had cardiac consequences that have already been discussed. And sadly, almost 20% of this study had unfortunately died. You can see a table that represents some of the findings in TANGO2 deficiency disorder, including speech and cognitive delay in nearly everyone, hypothyroidism and seizures in about half of the patients. We began the study really trying to understand the symptoms, and I think we have a better grasp of this now. And I think it's our community that is labeling these symptoms so we can understand, because it is quite difficult to describe the symptoms that go along with TANGO2. And I want to distinguish the spells from the crisis for those of you that aren't used to having a patient like this. That spells are really more transient symptoms. They describe loss of balance, falling over, extreme lethargy, head tilt or lag, you can see in the bottom, drooling, worsening of speech, staring off. But this needs to be distinguished from a true metabolic crisis, where the symptoms are the same and typically worse, but they're associated with laboratory abnormalities, which we've already discussed today, which include elevated CK, rhabdomyolysis, sometimes hypoglycemia, and EKG findings. I'm going to highlight again different individuals. And again, I didn't know that, Debbie, you were going to introduce me, but I want to say a huge thank you to Robert and your family, because Robert was one of the first that I interviewed, or Debbie was. And Robert, this is Robert on the left. You can see he's jumping up and down. He's the littler one. And this is Robert during a spell, and you can see the marked difference in the symptoms, and these can resolve, and very difficult for physicians to make this diagnosis, but even more difficult in a child with 22q11.2 deletion syndrome, who can also have difficulties with speech, with balance, with cognitive deficits. So Robert not only taught us about TANGO2 spells, but he introduced us to how important this is in terms of 22q11 deletion syndrome. For those of you, just as a reminder, TANGO2 requires, as you know, deficits in the TANGO2 gene on both alleles. It's estimated to now affect at least 8,000 individuals. That's from our more updated information through the Broad Institute. About 1 in 500 are carriers for this gene. A gene variant that is pathogenic, 1 in 300 in Caucasians, and 1 in 600 in Hispanics. Importantly, the TANGO2 gene lies within the 22q11.2 region. So children that have a deletion, meaning a larger chunk of DNA that are missing that constitute this syndrome, are also missing TANGO2. So if they happen to also be one of these common carriers, then they too are going to have not only 22q11.2, but also TANGO2 deficiency syndrome. And so we felt it was important that we be able to recognize these children. And through the work, there's work ongoing as an offshoot of the natural history study with Grayson Northcutt, who is a genetic counseling student here at Baylor, who's being led and mentored by Dr. Leilani and Mackenzie and some of our genetics team here. And he's going to review over 700 cases of 22q11 syndrome in our hospital, and his goal is to help determine how we can identify signs and symptoms that can distinguish TDD among those with 22q11. So his work is forthcoming, but I also want to mention that's not just where Robert ends. Robert, as Debbie mentioned, is now an angel, and his cardiac crisis is what we really also have been working to better define and understand, not only through this study, but as you've seen through some of the basic work going on. I'm going to give another highlight that Robert, and not just Robert, but so many of those that gave information through the natural history study is really what helped me help my own patient. This is Dagoberto and his family. So Dagoberto presented to my hospital, like many others, in acute crisis. He had significant heart failure, and when I begged to put him on a heart-lung bypass machine called ECMO to save his life, I was told no, that he didn't have something that was reversible. So he ended up with a cardiac arrest. You can see his rhythm. This is not compatible with life, but thanks to the natural history study and knowing what things didn't work acutely, it's what led us to use a new medication, isoproterenol, which actually is contraindicated with someone with such profound QT prolongation and tachycardia. So this is an unusual treatment, and I think it's a breakthrough. It doesn't work in all. I don't think it works sustained, but it is something that we now have in our back pocket, and I think it's an important finding from the natural history study. And I am grateful to everyone for allowing us to obtain that data. In addition, we were able to identify what we call a pathognomonic TANGO2 ECG. And that means that there are specific findings on EKGs that, to me, identify a TANGO2 patient, because during health, it's normal, just like Dr. Meisner mentioned, but there are specific changes that are very, very unique. And to me, that's another breakthrough. So how does this go? How does this relate to the clinical relevance? Let me summarize it. Number one, we've identified what we call in medicine a pathognomonic, a very unique EKG that is very important, because if the diagnosis is not made yet, but a patient comes in with rhabdomyolysis and this EKG, I think the diagnosis is there and they can begin treatment that is atypical, but should be used in a TANGO2 patient in crisis in arrhythmias. We've also identified an option for acute treatment of arrhythmias. And I think if the patient is in severe heart failure, we've been able to identify through the TANGO2 natural history study that the heart can recover. And this means that ECMO is now an option, and it is an option now. And I think that that's changed, at least at our institution. But really the question is, can we just avoid getting to this point? If this is so dangerous, how do we prevent this from happening to our patients? And let's go back. I'm going to go back to the natural history study again and how this has helped us. So in 2019, I told you we only had 22 children. And at that point, these are slides, actually information taken directly from slides from the family conference, the first one in June 2019. And at that point, we only had 7 TANGO2 genes. We now have identified 27. At that time, and you can see some of my slides, I was trying to figure out why some patients had crisis and others didn't. Why were some people doing better and others weren't? And what I found is that among the children that we had in the database at that point, they were on 36 different medications, including vitamins and supplements, because lots were thought to have mitochondrial disorders, so they were being put on lots of different mito cocktails. But what I noticed is that those that were doing better, and this is information that was given to me by families and just by doing a lot of interviews, those taking vitamins, regardless of not always, different brands, seem to be doing better. And I have to tell you, this is really what I look like. I really was scratching my head. I didn't know if I should even introduce this information in 2019. So if you go back to my slides, I did actually mention it, but I was very cautious about it. And I look back at my slides now, it's interesting that I noted that only 25% of the children at that time were either on a multivitamin or a B complex. And this was our conclusions in 2019, that crisis occurred despite certain supplements, that frequent feeding was important. And this was what I wrote: vitamin B or other nutritional supplements may be helpful, but we need to look more carefully at this before coming to conclusions, and further studies need to be performed. And so I'm very grateful that by 2022, we had 73 children enroll in the study and provide their information that was ultimately used to publish and provide the graph that you already saw. So for the first time, we had enough patients where we could do statistical analysis that showed us that children that were taking, so the green bars represent you're taking these supplements, CoQ10 and carnitine, but the green bars are showing that you're still having crisis. But those patients that were taking B complex or multivitamin were not. There is one patient that did have a crisis. That patient, we did not include TPN in our analysis. And so this is another important point we'd love to highlight. I'm trying to get more information about that subject. So there are, obviously, this information is being tested in multiple different labs. I don't want to take away from their thunder, but I'm hoping that you'll get to see more information about how some of the data coming out of the natural history study is being tested by investigators. I may not have everybody in here. Like I said, Dr. Mackenzie's lab is now doing it in C.elegans. But I also want to highlight that it's not just the basic science, that it's the families that are important in the natural history study. And it's important to highlight where the findings that we are seeing are actually making differences. So I'm just going to highlight a few that came off the Facebook site. So Annalyn's family says that she has made huge progress since leaving the conference last year and starting vitamins. She's talking more and more. Her life has dramatically changed for the better. Phoebe's mom says that the difference the new B vitamins have made so far is astronomical. She has gone from walking 50 meters to running all day, no wheelchair needed. She has also stopped having her TANGO2 spells every morning. Long may it last. And John's father says, since taking the B vitamins, John's energy has improved a lot. He's been playing soccer for over 2 hours now. The power of the vitamins are awesome, and we're very thankful for the foundation. Pictured here is also the first time we've gone to the beach, and John was able to enjoy the ocean and walk on the boardwalk. So what's the clinical relevance? Well, the natural history study has led to the information that taking daily B vitamins or multivitamins containing B can prevent metabolic crisis. At least one or more of the B vitamins, were still to be determined, seems to improve symptoms, energy level, strength, walking, maybe speech. Again, these are details we need to still understand. And that it's extremely important to not miss B vitamins, as this can lead to crisis. But how much and which B vitamins should I be taking? When should they be started? Is it just B5 or is it others? Or is it a combination? Is it possible we're taking too much of one and less of another? These are all questions we're still trying to answer. But in terms of when should we be starting, maybe we've actually found some potential answers. And that answer to me came during the last conference in Orlando in 2022, when I was introduced to Seth. This is Seth Harris. And as you can see, Seth walks completely normally. He attends school. And when I talked to Seth and had a conversation with Seth, it was like talking to any other teenager. And I was so shocked that it really sparked a complete change in some of my thought. And the first thing I asked his parents, you can imagine, is Seth on a B vitamin? And they said yes. He started B vitamins at 9 months of age. So he was our youngest who started B vitamins. I gave Seth, and this is part of the natural history study, a developmental profile test. It's a very kind of short 20-minute test that asks some questions. It's not in depth, but it gives you kind of a general score in 5 different areas: in physical, adaptive, behavior, social, emotional, cognitive, and communication. You give each a score in each of these, and then gives you an overall general developmental score. Preliminary data showed that those on B vitamins in our study have higher general developmental scores, with Seth having one of the highest scores in our group. This is also preliminary data, but I just looked to see what's the difference in this DP4 score, depending on when you started taking vitamins. And this is just kind of prelim data. I don't have any p-values for you yet because I don't have enough patients or subjects. I need to do more. But you can see the bar represents what's average score in the population, age-based score. And you can see that there seems to be a difference, and perhaps there is a difference depending on when you start taking these vitamins. So I want to thank Michaela, Seth. This is Seth, Michaela, and his sister, Michaela. And this is important because Seth and Michaela share the exact same gene. And yet Seth has really not minimal or no symptoms, got started early. Michaela's doing great, but she had multiple crises and wasn't diagnosed till later, did not start her vitamins until she was a teenager. I want to highlight another family because the Pochettinos, Eduardo and Carlotta, have always been in our study, but it takes more than one patient to be able to identify something very important sometimes. And Eduardo had crisis. He's the older brother. They share the same gene. And you can see Eduardo's doing much better. He got started on vitamins in later in toddler years, but his sister got started more in infancy. And Carlotta, you can see, runs, she jumps, she acts like any other normal girl her age and really has full control. She doesn't have any signs of TANGO2. So now we've got 2 families whose children she started vitamins early, do not have any signs. And I have to tell you, this is data. I showed it to you before from the natural history study. It was hard for me to understand why not everybody had all the symptoms. But now we're beginning to understand possibly early identification and diagnosis, like Debbie Deloche is probably working on, could make an absolute difference if we were able to get treatment started potentially earlier. So that answers the question. I don't think we can say with just 2. So this is more work that needs to be done. And I'm hoping that more people will enroll and we can truly answer this question statistically. So overall, how has some of the data from the natural history study contributed to medical information and clinically relevant information to the medical community? We've updated the emergency letter, so if you don't have a new one, please look. The data is now updated based on the natural history study data. And we've also put on it a QR code where it provides physicians with the ability to go directly in to our more detailed update that's in Gene Reviews that's written by many different specialists. And it gives specific data about not only how to recognize, but manage it, during health, but also during acute crisis. And it does talk for the first time about vitamin supplementation. One of the things I want to bring to highlight here is this idea of a multivitamin banana bag or IV fluids with multivitamins in it. And I'll show you an example of why this might be important. A banana bag is used in adults who have alcoholic intoxication and are deplete of important vitamins. And so previously, or I don't know if it's still being used, but we used to hang banana bags when I was a student. And these IV fluids, when they came into the emergency room, contained the full complement of vitamins. And it includes all the B vitamins, which are important. And it has a yellow color too, which gave it the name banana bag. So I'm going to bring you back to one more important family. I don't know if they're on today, but I want to say thank you to them. And their team. So this is the Butler family, Addison and Caitlin. They just joined our natural history study yesterday. So the data is a bit limited, but I think the data is important to share. So Addison and Caitlin are sisters. Addison is 7 years old. They both have cystic fibrosis. And if you know cystic fibrosis, you know that they have a problem absorbing certain vitamins, particularly A, D, E, and K. So they're generally on replacements, but Addison and Caitlin have been taking multivitamins since infancy. However, in the past year, unlike her sister Caitlin, who's been really good about taking her vitamins, Addison has not been consistently taking hers. In November of 2022, her parents noted that she was more lethargic, falling over, having balance issues, worsening speech, cognitive issues that brought them to many physicians' admissions, and unfortunately, they weren't able to find the diagnosis. In September of last month, she had a severe episode, went unconscious, was noted to be hypoglycemic, had rhabdomyolysis, and was admitted to the hospital and was noted to have prolonged QT. Within days, she had 2 cardiac arrests. Her astute team at this point was recognized that this might be TANGO2 deficiency, but they didn't have the genetic confirmatory test to make sure it was. And despite all of their efforts to treat, her lab values, her CK and everything was continuing to worsen. And there was a lot of concern. At this point, I got several calls, as has happened many times in the past. But this team actually gave their cell phone, and I was left clinic in order to talk to them. And what they did really impressed me, because based on her symptoms, I told them that I thought almost positive that she had TANGO2 deficiency disorder. And we came up with a plan, which I've shown on the right. So Addison is one of the first that were really utilizing this plan. And it's very, I think, unusual, and it would be hard for others to potentially implement, because when you're hypoglycemic, it's hard to listen to me say, decrease your dextrose. Decrease your IV fluid rate, even though you're in rhabdomyolysis, because that's not what we normally do. Discontinue Precedex, which is an anesthetic that's often used to help when someone has arrhythmias, but you don't want to be bradycardic with this. Discontinue your lidocaine. That's the major treatment for long QT, antiarrhythmic. That's hard to do. And give vitamins. IV folate is one that is available. So we were giving anything that they had IV. And we told them to try this banana bag and to start TPN, to get whatever vitamins they could into Addison as quickly as possible. And this team did it. I'm happy to say that Addison started to turn around. Her CK dropped. Everything turned around. She's now recovering. She's out of crisis. And they're weaning off the TPN. So I want to thank Addison and her family for allowing me to share this story, because if I didn't, you wouldn't, be able to hear this information. Some of these patients being able to hear this information kind of leads us to further studies, whether it's clinically or whether it's in the basic sciences. Many of you are taking this information. So again, I think our hospital now has this on our, we can make an order, a TANGO2 patient arrives in our ER, and I can order a banana bag immediately to initiate in the emergency room. So what's next? I mean, there's so much that we need to learn still from the natural history study. Information like, why do some have seizures and hypothyroidism? What's the best treatment? How do we treat seizures and stiffness? Do B vitamins treat these other symptoms? Will they progress, or can we halt them? Can we cure them? Why do some children still have symptoms even if on B vitamins? Is it that we're not on the right ones? We're not on the right combination? Are we giving too much B vitamins? What dose should be given? These are all things we're working on actively. We are actively working on it, but again, we don't have enough numbers. So, we're hoping to initiate a prospective natural study as a team. I think that'll help answer some questions. But in summary, my update in the past 4 years, the natural history study, I believe, has changed the history of this disorder, but we have so much more to discover. There's incredible power in every child and every family, and it's you making the difference. So thank you for being part of this study. Please encourage other TANGO2 families to enroll. This is the only way we're going to continue to find answers. So thank you very much.
If we're ready for the next speaker, hello, my name is Robin Mitchell, and I am mom to Hannah Mitchell, who is actually celebrating her 29th birthday today. It's my pleasure to introduce the next speaker, Dr. Elizabeth Ottinger. Dr. Ottinger is the acting director of the Therapeutics Development Branch in the National Center of Advancing Translational Sciences at the National Institutes of Health. She manages both the therapeutics for rare and neglected diseases and the Bridging Interventional Gaps programs that provide in-kind resources to help researchers translate molecules from bench to bedside. Welcome, Dr. Ottinger.
Hello, good morning, everyone. Thank you so much for the invitation to present here today. I'm going to be talking about paths to treatments, enabling preclinical therapeutic development for rare diseases, and to let you know how our programs are actually working with patient groups, industry, other government agencies in order to move molecules through the translational space into the clinic. Sorry, it's not advancing. I can't get it to advance. Oh, there we go. I have nothing to disclose and no conflicts of interest. And so as everyone here is aware, there's, the huge public health challenge of over 10,000 rare diseases and only 500 treatments. And the time from early development to a medicine can take 10 to 15 years. And 9 out of 10 of those that are in development often fail as clinical candidates. So in our therapeutic development branch, which is part of the intramural program of NIH, we try to address these challenges in developing therapeutics for rare and neglected disease. And we operate as a team of teams, which I'll show you a little bit more about after, where we have very strong project management and technical expertise to try to help our collaborators in their projects. And we foster close interactions among the clinicians, the researchers, and the patients, because as everyone knows, that's essential to actually developing a therapeutic. So this is a little bit about our portfolio. We have a broad distribution across therapeutic areas and modalities. So we're disease agnostic, but as you can see, we have we work in many different areas: CNS, pain, musculoskeletal, ophthalmology, and we also work in different modalities. And so our projects are distributed over small molecules, protein peptides, and gene therapies. And since the initiation of the therapeutic development branch, we've enabled over 52 INDs to support clinical trials since its existence, and 17 INDs in the last 4 years. And actually, we have one to add, which I'll tell you about in this talk, that we just received safety to proceed last week. So I need to update that to 18. And this is just a total of the 14 projects and collaborations that we've had over the last 4 years. And what I just wanted to show on this slide is that, again, you can see our portfolio is distributed over different therapeutic modalities, but what our real goal is to de-risk. And so we work with all of our collaborators to just keep moving these through the preclinical pathway into clinical testing for the patients. And so many of ours have gone on where they've either partnered up or they were already a company that came in to work with us. And so they've gone on and work on the molecule into the other phases of development, the late stage development. So we really take a team of teams approach to the way that we foster therapeutic development. And what I have here on the left is you can see the types of projects. We really try to develop our portfolio so that we can actually try to address more than one rare disease at a time, if possible, using platform approaches, finding disease commonalities, if there are novel mechanisms of action, improvement to the current standard of care, alternative treatment paradigms, drug repurposing and molecule repositioning is also a big area that we've worked in some of our projects. And we really just want to advance new technologies and drug device combinations also. And so on the right, what you can see is this is really our model at our therapeutic development branch. And what I was telling you is that we really value project management as a very critical key component since we started the program, because you really need those champions who can drive the project. There's so much always going on within a project, the different studies that need to be done, how you're working with the different external collaborators, or, because we're an organization that works both doing some studies in-house, but we do a lot with external CROs that really has to be managed really well. And we have a series of, which I'll talk about in a minute, disease expertise areas in the drug development process. And so again, all of these different partners and functionalities, they need to be coordinated well so that you can actually keep the project plan going through its execution and making sure decisions are made based on the data and that time, budget, resources are allocated well. And so again, what we utilize is this central project management that then moves things through the preclinical development pipeline. So the area many of you may have seen before, this is a chevron diagram that's often given to kind of define the phases as you go through preclinical development. And so you'll have things in, target discovery where you're really trying to develop your assays and develop a molecule, your identify your candidate. So our space in the therapeutic development branch is really at that lead op to preclinical space to move things through the regulatory FDA studies that you need to support your molecule for clinical testing. And so what we have is, again, we start with that clinical candidate normally, and sometimes we often do lead optimization for our partners that we work with, but the beginning stages is really that proof of concept studies that the molecule is working and is really affecting the disease. And as you've heard from different animal models or 3D models are now also being utilized to support in some cases at least efficacy. So you start at that candidate selection, and then there are a series of areas within your preclinical development, as I've outlined here, that you really need to address with that molecule, your lead candidate, through the different parts, like your distribution and your pharmacokinetics. How does the drug, metabolize and go through the body? What are the concentrations? The CMC, how you actually manufacture it. You need to make larger scales, and it has to be formulated. You have your safety and evaluation in your animal models, your toxicology, so that it's safe to proceed and that you can actually start your clinical trial. And all of this is at the end, you need regulatory submission in order to put all of that preclinical package together so that you can actually enable your IND and start your clinical studies. And what I wanted to really introduce was the way that we actually base our projects is we use a target product profile. And so this is something that you'll often hear in industry and in other aspects when people are doing drug development. It's really where you start with the end in mind. You think about what you would like this molecule to look like when you're actually testing it in the clinic. So you have different areas, and often this is correlated to the labeling of your drug. How are you going to utilize this drug for patients, and how is it going to actually be efficacious? And so you have to think about these different parameters in your target product profile, and then you have to think backwards into your preclinical development program, and you have to design all the studies that are going to support what you're hoping your end product will look like. What is your target product profile? And so if you see on the right here, I'm not going to go through everything, but it's just a way for you to really think about what studies need to be done in order to support your preclinical development program. So this is a very key component of our drug development process in the therapeutic development branch. And so I'm going to give you one case study. This was a drug development for a disease of an unmet need. It's Jansen's metaphyseal chondrodysplasia. It was the development of a peptide, an inverse agonist for the receptor called PTHIA. And the disease manifestation is that it affects bone development and mineral ion homeostasis. You have increased calcium. And this is often a pediatric disease, and these children often have to undergo many surgeries in order to try to correct the bone deformities. And it's very painful, and these children really go through a lot. This project came to us. It's a collaboration with Mass General Hospital and Harvard Medical School with Dr. Harold Jupiner and Tom Gardella. And they had a proof of concept in an animal model. So when they were doing twice daily injections of this molecule into a mice model of the disease, we can see here on the right, well, in the red there, when the actual drug was given compared to the one next to it, you will see that it actually seems to improve the bone morphology. And so what did NCATS do? Well, this project came to us, and it came through a patient advocacy group, actually, kind of coordinating with the Mass General and making us aware and applying in with Mass General as the PIs on this program. And so we did all the IND enabling studies, and this is the one that actually just received Safe to Proceed last week. And so now we're in the process of developing the clinical trial. But we did, and we also supported a natural history study that was done here at the NIH Clinical Center. So again, it's about building a collaborative ecosystem, because it really takes this team of teams, like I was mentioning. So we had the Jansen's Disease Foundation. And again, those are our key connecting partners that enable the clinical research. We had our TDB preclinical project that had come through us through researchers and clinicians at Mass General Hospital, who had a molecule that they thought, had a possibility that looked like it was efficacious in some animal models. And then we utilized our NIH Clinical Center in order to do the natural history study and now to actually execute the clinical trials. And what I always like to emphasize that any of our programs start and end with the patients. So you can see here, this is kind of mapping through, I said it started with outreach, there was academic research. They came in and to the therapeutic development branch as a project, working with us for all of the preclinical studies that were needed. There's clinical planning, so the natural history, and now we're at hopefully the phase 1/2 trial that can start soon. And what you're all hoping, and everybody's hoping, is that this leads to the treatment for the potential benefit of the patients. So again, it starts and ends with the patients, and we always keep that in mind in all of our programs. The second case study is a platform approach. And so in this project, it's a platform vector gene therapy approach. And so we have this hypothesis that we're trying to test out at NIH is, if we use this platform approach, will this increase efficiencies in the preclinical testing and the clinical trial startup? And you can learn more if you actually go. There's a PageGT website. But the development of this platform, or the framework, is that we're studying 4 different diseases: 2 organic acidemias, which have the genes PCCA and MMAB, and 2 congenital myasthenic syndromes, which are CoQ and DOX7. These are collaborations with our other institutes. We're working with NHGRI, Dr. Chuck Venditti, and NINDS, Dr. Karsten Banneman, for the congenital myasthenic syndromes. What we're trying to do is really harmonize the processes using the same manufacturing processes, the analytical methods, the study designs, the utilizing full IND enabling studies, the regulatory, and have these common clinical protocols also when possible. And so what we're really hoping is that if we use these common processes and we make the data publicly available, that this is actually going to be able to catalyze for other groups the development of gene therapies. Oops. And so again, as I was saying before, everything is a huge team effort. And so we've had all these different institutes a huge team, over 30, 40 people, sometimes on a call, and you have to coordinate all that. So we have really strong project management, again, trying to move this through. And here's the current status of these four. We just finished a pre-IND meeting on the first candidate, PCCA. So you can see that molecule or that gene therapy, it's farther along in the whole regulatory pathway and the preclinical development. But what we're hoping is that if you have these example where we have everything kind of open, and that we can see what did we do in this first one, we can start applying that to the next ones that come along and find those efficiencies that will make it easier. Even if someone has a protocol, right, you can actually see what type of assays need to be developed. What did they do? Those are the kind of questions that we hope this helps to answer for others who are doing this for their rare diseases. So this is our regulatory path. And so we always believe that you need to interact with the FDA as early as possible. And so where we started was we had an interact meeting with the FDA. And this allows you, once you pick your preclinical candidate, you can go there. And we had some lessons learned even from this. I don't think we had enough CMC data. If we had had a little bit more about how we were thinking about and had some data of how we would actually manufacture this molecule, I think we might have gotten more out of it, although we did get a lot of information. We're in the process of writing that up, and then we will also share our Interact package with everyone. But what we have shared so far is we did get orphan drug designation, and we got rare pediatric disease designation for this molecule. And all of that material that we published, and we actually published the packages. And we tried to provide a guide for everyone. It's also on the PaveGT website so that others could utilize this as a learning tool and a guide for their therapeutic development projects. And so again, it's all about connecting with the researchers and clinicians. They also attended our FDA meetings. They were at the pre-IND meeting. We had patients there, and engaging in the natural history and clinical, studies. So again, it's patients being involved, coordinating and working with us in the development of these molecules. And as I mentioned, one of our goals is dissemination. Can we provide this open access playbook, to really let everyone have the experience of the know-hows and the lessons learned from implementing this platform gene therapy approach in all the areas, preclinical, regulatory, and clinical. And this is complementary to another program that you may have heard of that NCATS is part of, which is the Bespoke Gene Therapy Consortium, which is also working with FNIH, the Foundation for NIH, and other industry partners to also be able to understand gene therapies in different areas and different diseases, and they've announced which programs that they're working on also. And the last example, if I have a few minutes, is I was just going to talk about, I think all of us are trying to find ways speeding up the search for solutions, right? How can we, maybe one of the big challenges is data, and how data is in different places. And it's sometimes how do you bring that together to actually gain insight and knowledge that's going to help you find a possible therapeutic or other information about a rare disease. And so we tried to think of an initiative where we could actually hopefully make a contribution to rare diseases that would help both, patients, researchers, clinicians, that they would have somewhere that they also could go and try to connect, where we've tried to connect some of the publicly available data. And so what we developed, it's in development. So there's a lot more that we still need to do, but this is called RareSource. It's an integrated bioinformatics resource. And what we hope to do is that eventually, as we keep improving and building on it, that it will eventually provide a source of novel insights into rare disease commonalities through this data integration. And so we wanted to create a searchable, accessible, user-friendly interface, and that it's actually publicly accessible. And what we're trying to do is train the platform both through manual curation of disease literature and then integrating known databases and developing models for automation of the mining of the literature and bringing other data together and expanding that to the many rare diseases. And so if you'd like, the website is live. We're about to release our second version of version 1, which has them updated on the interface for searching. And so I just wanted to show you quickly what it looks like. So the first part that we really focused on was links to the literature, because we wanted to try to understand what data is missing in the literature that may get kind of lost because there's so much, different articles. And could we bring that together and do some literature searching? And so could we link the genetic etiology and the associated genes with the diseases and see how that would be in the literature AI? So we have a literature AI function. And so if you look, you can browse and you can go to the literature AI. And what it allows you to do is you can put in a gene and you can put in or the disease, you can look them up both separately. And what it will provide is like, where are a lot of the articles published? And then what's the publications, looked over the yearly trends and links to the articles. And so this allows you to then pick which articles you might want. And you can export that. And so you can search with rare disease names, and you can search with their associated genes, and you can do the co-occurrence, like genes and diseases or diseases and genes. And we're trying to use literature AI, things like chat GPT, trying to be able to pull out those articles. It's continually updated, that would have those references to that, that it would actually make those associations. And so, and part of it also is that there is the gene structure. We have 3D structure in there. So we've assembled a lot of information, also using things like AlphaFold, which are the AI, where you can actually get structure. So there's a different, again, data sources that come together to show the structures, and it shows where the variants are. There's a lot of information there that you can actually click on and see where the variant is and what part of the protein it is and what part. So, and this is a team where we work with NCI. They have an Advanced Biomedical Computational Science Center, and we've been working closely with them and with NICHD and other partners across NIH that we're trying to continually update. And so you'll see more versions. We hope by early next year, we're actually going to have all the variants curated, and also curated out of the literature and using AI tools. And so hopefully that will make nice connections between the genes, the diseases, and the variants. And then again, hopefully in the future, we hope to have more natural language processing where you'll be able to get more information that can associate phenotypes or other types of information with the genes, diseases, and the variants. And I think that's where I'll end. I just wanted to give you some case examples, how we work in therapeutic development, targeting a particular molecule, and how we work more in the broader sense, trying to advance rare disease therapeutics and information for the rare disease community. This is our group. There's about 55 of us. So it's a small group, actually, when you think about it. But we do work with so many collaborators and so many extramural partners that our group actually and our, is very large. And so I appreciate everyone's time. And this is just, again, NCATS, and I probably should have put, but hopefully you get the rare source link, and I'm open for any questions. So thank you for your time. And I hope this was somewhat helpful for you to see a little bit about a program at NIH that is trying to move towards therapeutic development and helping rare diseases. Thank you.
Hi, everyone. My name is Cesar Martinez, father to John, who lives with TANGO2 disease. I am happy to be part of this event today and proud to introduce our next speaker, Dr. Sam Mackenzie from the University of Rochester in New York. Dr. Mackenzie is a clinician scientist focused on gene-based therapies for neurogenetic disease affecting children. He spends his time taking care of children with neuromuscular conditions conditions and is also actively involved in basic science and transitional research in the lab. Dr. Mackenzie is also the chair of the TANGO2 Research Foundation Scientific Advisory Board and active member of the Foundation's Research Committee. Dr. Mackenzie.
Thank you very much. And let me see if I can take over the screen here, request remote control. I decided to give you an honest picture of what I look like most days, rather than a polished headshot. All right, let me just advance here. It's like I can. Perfect. So I'm really thrilled to be talking to you today about our human subject project in collaboration with the natural history study run by Dr. Lalani and Dr. Miyake that you heard about a little bit. And this is sort of framed in the setting of a transition as we think about a prospective study. So that's been kind of one of the themes today as we've moved from the morning talking about basic science. How do we measure disease features in patients, in kids, in young adults? And specifically, we're going to talk about movement disorders. So none of my conflicts of interest here have any relevance to the talk. And this work was funded through a grant through the TANGO2 Research Foundation. So just to acknowledge this group. So overall objectives of the talk, what I'm going to talk about today, we're talking about movement disorders. And I think this is a common point of confusion, certainly for parents. You know, what's a spell, what's a crisis, what's something else, what's none of the above, and even clinicians. And so I will go so far as to say even pediatric neurologists can be confused about what movement disorders are or what to label certain things. So we're going to get into the weeds a little bit on the movement piece. Specifically, I want to talk about dystonia and ataxia and what those look like. And then we're going to get back to this notion of TANGO2 spells, these episodes of fatigue, unresponsiveness, and how those might differ from the dystonia, ataxia, or other movement disorders that we can identify. All right, so just a quick background. If we think about movement disorders, we're really talking about a specific, what we call semiology, or a way to describe the movement. And you can break those down into two kind of broad categories. There's decreased movement, which those are pretty rare. And then there's increased movement, or we call hyperkinetic movement disorders. And you can see some of the examples here. This is not an all-encompassing list, but just to show you dystonia, where there's increased muscle tone kind of transiently most of the time is an example of that. Or chorea, where you see these large amplitude movements, not so much a common feature of TANGO2. And why is it important that we even talk about movement disorders or do this work? Well, I think, as we've heard, sometimes that's the presenting sign, right? Sometimes parents will notice, hey, you know, why is my kid leaning to the side like that? Or why is her neck flexed like that, looking upward at the ceiling? And these can be really confusing. And then you go to your pediatrician and they say, well, they look fine today. Let's see if that happens again. And that's a missed opportunity, I think, to make that diagnosis early. And we've already heard some of the reasons why we think early diagnosis is so critical in TANGO2. So that's I think the paramount reason for really putting a name on some of these movement disorders. Secondly, you know, I think it tells us a little bit about what's going on in the disease, right? So I'm going to show you, again, highlighting the dystonia and the ataxia, and talk a little bit about some of the brain regions that are implicated in those movement disorders. And so that tells us, you know, maybe we should be thinking about what's happening in those brain regions, what's happening in the cells that live there, what sort of neurotransmitters do they use to communicate with other cells in the brain. And that might tell us a little bit about what's going on in TANGO2. And then, you know, we've just heard about, you know, gene therapy and we've heard about vitamins and maybe there are other non-gene therapy treatments that might beat vitamins that we need to be thinking about. So as we move towards an era of therapeutic clinical trial testing, right, we need to be able to measure TANGO2 features in a way that's quantifiable and rigorous and precise. And it's a challenge to do that in a disease like this with so many different features where kids don't necessarily present the same way. So I think starting to delineate and get a little bit more granular about what these features are, what these movement disorders are, is critical for any sort of testing of therapy. All right, so I've shown this slide before. This is just kind of how I think about the general gestalt of research as it relates to any rare disease, but certainly for TANGO2. So on the left here, you can see, sort of the patient side of things where we look retrospectively what's happened in children's lifetime. We can take that into a prospective natural history study, which, that's a big hope for the foundation, I think, is that we can get there in the very near future. And then we think about what outcome measures might we want to look at in that prospective natural history study that then could be parlayed into clinical trial. All right, so what do we do? What do we know about movement disorders and TANGO2 deficiency disorder? So we know that they're pretty common, right? And there's been a lot of case series that have been published. There was the retrospective NHS that shows a lot of kids have movement disorders. Now, the problem is when we go back to the literature, we're kind of relying on certain clinicians to make that call. Is this a dystonia? Is this an ataxia? Is this something else? So the terminology and the inter-rater reliability of what those episodes might be is a little bit in question. We know that they're variable. Not every kid has them. We know that they don't occur all the time. They're hard to catch, right? So that's a challenge. Certainly, if you can't bring a kid in any given day of the week to see what's going on, we're relying on things like patient videos. And we know that they may occur during metabolic crisis. They may not. That's, I'm sure, a huge question for many on this call. If your kid is showing some features of dystonia, what do I do? Do I need to take them to the ED and get labs drawn? As I talked about, this might be the first thing that shows up in some of our kids with TDD. And just to hammer it home, these are not readily identifiable by really non-specialists a lot of the time, and they can be hard to kind of call what's going on. So this project was really born out of that challenge, and we had two aims going in. We wanted to really just get a better handle on what these movement disorders are, what these abnormal movements are more broadly. And then secondly, think about the context in which they occur. So are there anything that triggers them? Are there anything that makes them better? What ages did these happen? And how often did they occur? That sort of thing. So I'm going to start with aim one here and get into how we approach this problem. So I've talked to you a little bit about some challenges, just to add some more to the list. This is a pretty rare disease. We're fortunate to have such a great network here. And so that's really helped with subject recruitment. But people are all over the world. These don't necessarily occur on a daily basis, as I said. So what we're really relying on here is a remote assessment, and we're relying on sort of a cross-section in time that hopefully gets captured on an iPhone or whatever your phone company of choice is. I don't have any disclosures related to Apple. And we wanted to take this to kind of the highest standard of review. And I mentioned that not every pediatrician might be familiar with what these are. And certainly not every child neurologist might be familiar with what they are. So we actually brought in people that do pediatric movement disorders to take on some of this review responsibility. So I'm showing 4 people here. 3 are at Rochester: Dr. Jen Vermillion, Angela Hewitt, Jonathan Mink, and then Miriam Hall, who's at Texas Children's. And so our schema kind of here on the left is what we relied on a lot of people on this call to send us videos of their kids, maybe having an abnormal movement, or maybe a video of them kind of in their normal activity. And we gave them a link to a HIPAA-approved box folder, which was secure and lets our experts review what these are. And then we basically just kind of classify what we're seeing in the video. AIM-2 here, we haven't actually launched into because we've been still on the subject recruitment and characterization end of things. But AIM-2, we're going to go back to the families that submitted videos and ask those questions about context and hopefully clarify a little bit about when these might occur and what might make them better. And so to give you an update, this isn't even up to date because we had a couple more families submit videos just this week. So we now have 25 videos or 25 TDD patients that have submitted videos, and we've reviewed 18. And basically, what we've tried to do is split these into what we call a primary movement disorder or a secondary movement disorder. And it's a little bit of a false dichotomy, but primary, we've said, okay, what's a common theme across all these videos that might have been submitted? What's the most consistent? What's the most obvious? That's clearly the most commonly occurring, and that's what we're calling primary. And if we're seeing some features of another one, but it's not necessarily that most in-your-face semiology, we've labeled that as a secondary. And kids could have more than one secondary movement disorder. And so we've had all of our experts review these individually, and then we come together for a consensus meeting, watch the videos one more time. And we've had really nice consensus in terms of what's going on. So out of our 18 that we reviewed, 17 had abnormal movements. And of those 17 abnormal movements, we were able to label the movement disorder in 15 of them. And I'll get into a little bit about what we saw. We had one subject where the parent right off the bat said, well, I don't have any videos of abnormal movements, but here's some other videos that I've taken that kind of illustrate dysarthria or trouble pronouncing words. So really, I think this is a higher number than what we've seen in the literature, which I'm not surprised by. It's a little bit of a ascertainment bias. If we're asking for abnormal movements, we're probably going to get more videos of abnormal movements. But I think it might hint at the fact that this is more common in TANGO2 than even we've appreciated in these case series, or maybe even the natural history study. So the two subjects where we didn't see a clear movement disorder that we were able to label, we did see these episodes of fatigue, decreased responsiveness, and we really didn't have a name for those. And I'll say, we even took some of these videos where we had consent to do so to a conference of child neurologists. In a specific session at that conference, we had a room full of the nation's experts in pediatric movement disorders, and I can tell you that they were stumped by what these TANGO2 spells were. So I think we are basically concluding that they are not a movement disorder. Something clearly is happening there, but I think that distinction is probably important. So let's get into what we saw, what we were able to classify. So we think dystonia is the main movement disorder in our small sample so far. We had 12 out of 18 where we saw dystonia as the primary movement disorder, and an additional 2 where it was a feature of the videos that we reviewed. And dystonia was commonly present in the neck. So we've seen the retrocollis, the neck, or the head arched upwards. Trunk, very common, which is the leaning that we're seeing. And then distal limbs, and this can be hands or feet. I'm going to show you a few examples of both of these. So here's one child with the truncal dystonia. And you can see that there's leaning there. He's not otherwise distressed by this. And I think we've seen some similar videos earlier of children walking with this truncal dystonia. And I'll show you one example of the limb dystonia here. All right, I succeeded in putting an arrow here. So a little bit of the bending at the knee. You're going to see inversion of the foot. So see that foot kind of turns in a transient fashion. And that's basically uncontrolled, uninhibited muscle tone. We'll talk a little bit about that. And so dystonia, when we try to think about localization as neurologists, where in the brain this might be occurring, the key region is an area of the brain kind of deep below the cortex called the basal ganglia. And the basal ganglia is kind of like the brake for a lot of our movements. And so if there's some dysregulation of the basal ganglia, you might have sort of uncontrolled tremor or uncontrolled movements generally, but dystonia is when you have abnormal muscle tone. And if you don't have that inhibitory signal coming from the basal ganglia, that can arise transiently or can be more common occurring throughout the day. And so this is linked to several different neurotransmitters. The one that comes up most commonly is dopamine. And interestingly, I've had a few conversations with parents who have had children with dystonia, one of whom said that their child was started on carbidopa-levodopa and the dystonia resolved. Now, I'm not saying that as a clinical recommendation to start levodopa-carbidopa, but I think that's a really interesting observation that we really need to explore more, which is going to be part of the second phase of this work. If there are treatments for this dystonia, I think that could be a huge factor in quality of life. All right. So ataxia. Ataxia was seen as the primary movement disorder in 3 out of our 18. And then we also noted it as a feature in an additional 4. So about half of our kids. And here's a pretty prominent example of ataxia, which is really just unsteadiness. It's an inability to balance. It's you can see it in other features that aren't just walking. So reaching for things, any sort of target related task can show features of ataxia. And this is a pretty extreme example where he's just really unable to stand and walk. And then let me see if I think this one still had. Sec. So I forgot to turn the sound off. And you can see here, there's ataxia, but there's also that truncal lean, right? So this is probably some superimposed dystonia. And you can appreciate here where it can be a challenge on a short snippet of video to tease these out, which definitely, again, tells us that we need to do a little bit more rigorous work with a prospective natural history study in terms of collecting these data. So what is ataxia? When we think about brain regions and localization, it's the cerebellum, which is in the back of the brain. And this can be really increased error with anything like reaching for an object to maintaining a certain pitch with your speech. So people can be described as having ataxic gait, ataxic pitch, tremulous hands while reaching, eye movement abnormalities, and then the gait is the most common. And we don't have specific treatments thus far for ataxia, other than kind of getting at the underlying mechanism of what might be causing it. We did see a few other types of movement disorders that were more rare. Myoclonus, which is a non-rhythmic jerking movement, similar to chorea, but much faster and a different kind of localization. And then stereotypies, which are definitely not unique to TANGO2, but those are sort of pattern involuntary non-reflexive movements that can occur. They kind of co-occur as motor tics. You might have heard that. It's common to see those in patients with OCD or or Tourette syndrome, for example. And I didn't show any videos of the TANGO2 spells, but I think we've seen these in other presentations. And these are fatigue, decreased responsiveness, trouble with speech, kind of eyes closed, partial loss of tone. This was described by one parent, partial loss of tone, partial loss of function, clearly preserved awareness. We've had patients that have had EEGs done during these episodes, and there were no seizure activity. It was just kind of a slow background pattern on the EEG. And interestingly, we see that there can be features of dystonia or ataxia that go with these TANGO2 spells. But we think that the TANGO2 spells are unique and that they might occur on their own. And again, we don't have a specific label, a medical label to say what those are. So, I talked a little bit about the second aim, and we're really excited to launch into this. So that'll be the next phase for those parents who have been involved with this project. Expect our team to be reaching out and try to get a little bit more data on specifically what was happening around these videos that you sent. Do you think we're right about our assessment of what we see? And mostly, I think most importantly for patients, if there's anything that you've identified that have made them better. All right. And we're also very excited to kind of think about these movement disorders in a quantifiable way. So we're going back through some of our videos and seeing if we can extrapolate sort of severity of dystonia, for example, as a quantifiable outcome measure. Think about how often does this occur, how long does it occur, and then how impairing might the dystonia be, where does it occur in the body. Anything that we can, again, quantify is something that we can use as ammunition in a clinical trial, because that's something we hope to reverse. And just to give a quick note of thanks to everybody involved in this project, particularly Drs. Vermillion, Hewitt, and Mink, and then all of the families that submitted videos, as well as Amanda and Jordan Taggart, who were instrumental in kind of providing input at the outset of this project, and certainly the TANGO2 Research Foundation for the funding. All right, and I will take questions.
It's my pleasure to introduce Dr. Michael Sacher. He is a professor in biology department at Concordia University with an adjunct appointment in the Department of Cell Biology and Anatomy at McGill University. His lab was the first to show benefits of vitamin B5 in flies and human cells. He is also a grant awardee of the TANGO2 Research Foundation. Welcome, Dr. Sacher.
Thank you. Let me just get control here. See if that works. Working. Good. Okay, well, so I was thinking as I was, my slides have a mind of their own here. I was thinking as I was just getting introduced here that there's, as the last speaker for this symposium, there's some advantages and disadvantages of that. The advantage is, I mean, that you can look back at all these talks, great talks, and I think a lot of complementarity in the research, and it seems like ideas are sort of converging, which is a good thing. The downside of being the last speaker is that because these talks were great and the background that was given to each talk was great, it feels like my introduction is going to be pretty much redundant, and so you'll have seen a lot of this before, and hopefully you'll bear with me as I go through that. So disclosures: I'm a member of the Scientific Advisory Board and the Board of Directors here at the TANGO2 Foundation, and also have received funding through the foundation to do some of the work, and especially the work which I'm going to show you today. Right, so I'll give you a little bit of a background, which, like I said, is going to look very familiar to some of the other, to a combination of some of the other talks. Then I'll get into some of the data that we've obtained using Drosophila, that fruit fly model system, the vitamin B5 data. Some of this has been published, and I'll show you some unpublished data towards the end of the talk, which includes some lipidomic analyses on human cells, which are deficient in TANGO2 directly from patients. And then I'll give you some conclusions and what we might be heading, or where we might be heading with this afterwards. So what is membrane trafficking and why is it important? To start with membrane trafficking, because that's really where TANGO2 has its origins. As Agustin talked about in one of the earlier talks, TANGO2 was discovered as a protein that may have a role in this process of membrane traffic, which really describes the movement of proteins and lipids between various compartments within a cell. And so these lipids, these proteins, can be carried in small vesicles that pinch off of one compartment and fuse with another compartment. Okay, so generally we have the whole process of delivering proteins from one compartment to the next. And my lab focuses on the endoplasmic reticulum to the Golgi, which is one of the first steps in protein secretion. And so you need to have vesicles that bud from a compartment. They need to be somehow guided to the appropriate compartment. They have to be first loosely tethered to that compartment. And then at that point, they can actually just fuse and deliver their content. So many of the proteins required for these steps were known, but in 2006, a paper was published which identified what are now known as these TANGO genes, which, as you've heard already, stand for Transport and Golgi Organization. And it's important to note that they don't necessarily have anything to do with each other. So just because we know the function of TANGO1 or TANGO3 or 4, or whatever we know the function of, doesn't necessarily mean we know the function of any of the others. And TANGO2 is the subject of what we'll be talking about, or what we've been talking about all morning and into this early afternoon here. There are a number of mutations since 2016 when the gene was first linked to human health. There have been a number of mutations that have been identified in the gene, the most common of which is a deletion that encompasses exons 3 to exon 9. And the exons are the regions of the gene that actually have the instructions to make the TANGO2 protein. And so if you're missing that large a chunk of the gene, obviously you will not be making the TANGO2 protein. And we're using cells derived from patients that have this particular mutation. There are other mutations that have been shown over the years, nicely summarized in a recent publication. One in particular is this 22q11 deletion, and that's shown on this slide over here. You can see it's a deletion of a chunk of chromosome 22, which encompasses the region where TANGO2 also sits, and there's a number of other genes that are deleted as well. And as you've heard already, then individuals with 22q11 deletion syndrome may also be prone to TANGO2 deficiency disease, TDD, if the other allele on chromosome 22 from the other parent has a mutation in the TANGO2 gene. So I think what we're finding here could be relevant to a subset of 22q11 deletion individuals. Okay, we've seen what the clinical manifestations are: developmental delay, speech difficulties, intellectual disability, pretty much for all individuals. Seizures are seen in some individuals as well, and I'll talk about that briefly when it comes to the fruit fly. And then the metabolic crises that we've been discussing, which are the crises which are the feature of the disease, which is can often result in mortality. And so we first got involved with TANGO2. I apologize for the background. I think I had a white background when I gave my slides in, but now I think things aren't showing up so great, maybe on the green. But we first got interested, when we were first interested in TANGO2, we were working on the assumption from that 2006 paper that it was required for transport between the endoplasmic reticulum and the Golgi. And so we took patient cells and control fibroblasts, and we performed a rather simple assay where we have a fluorescent protein, which is locked in the endoplasmic reticulum, and then we can release that protein whenever we want, and that fluorescence will move from the endoplasmic reticulum to the Golgi, and we can simply monitor the rate of increase of fluorescence in the Golgi over time. And that's what's being shown over here. So in control cells shown in black, you get a particular rate of increase of that fluorescent protein in the Golgi. And in the cells taken from this individual with a TANGO2 mutation, you can see that that's strongly delayed. And we know that this is due to TANGO2 because when we reintroduced a wild type version of TANGO2 into those patient cells, we see what the curve in red you can see is now pretty much in lockstep with what's happening in the control cells, suggesting that TANGO2 is what's responsible for that delay in trafficking between the endoplasmic reticulum and the Golgi. So we looked right away at what the Golgi looks like, because very often when a protein strictly functions in endoplasmic reticulum to Golgi transport, the Golgi morphology changes. And so what does the Golgi look like in a mammalian cell? Well, in blue here is the nucleus, and what we're showing here in yellow, and we've separated various channels down here because we looked at several proteins, mannose 2 and another protein called P115. And you can see this is what the Golgi normally looks like. It's right up against the nucleus, got this ribbon-like pattern. But for 3 different patients, we didn't really see any differences between what their Golgi looked like versus the control. And that was a bit curious because, as I said, almost every protein that functions in ER to Golgi traffic will do something to the morphology of the Golgi. So we looked at where TANGO2 might be localized in the cell, and you've seen some of this from Augustin's talk as well. We put a fluorescent probe onto TANGO2, and we put it either at the beginning of the protein or at the end of the protein. And what I mean by that is the protein is simply a string of amino acids. The first amino acid we could think of as the beginning of the protein. The last amino acid is the end of the protein. So we put our fluorescent tag either at the very end or at the very beginning. And what we saw when we put it at the end of the protein was that TANGO2 looked like it was give a pattern that looked like mitochondria. And when we stained the cells for a mitochondrial protein and then superimposed our signal for TANGO2 onto the mitochondrial signal, this is what we saw. And as you've heard before, and maybe you remember from, I don't know, kindergarten or grade school, that when you mix red and green, you get yellow. So we start to see, we artificially color one of them red, artificially color the other one green. And when we superimpose, we start to see yellow. So it looks like some of TANGO2 was localizing with the mitochondria. And curiously, when we put that tag on the beginning of the protein, we lost that mitochondrial signal, which suggested to us that there was something at the very beginning of the protein that was helping it localize to the mitochondria. And we showed that within the first 40 or so amino acids, there was some sort of determinant that was required to get it to the mitochondria. We haven't really pursued that. And so it looks like, okay, TANGO2 is on the mitochondria, but as Christina showed in her talk on the natural history study, those individuals on mitococktails do not seem to have a reduction in the incidence of a metabolic crisis. So that would be that was a little bit perplexing to us, that this mitochondrial protein not being that patients aren't responding to mitococktails would suggest that something different might be going on here. Okay, so given that, what I mentioned about the mitococktails, also we noted there's some similarities in the phenotypes between individuals with VLCAD mutations and also an ATGL deficiency, which I'll talk about a little bit later. But ATGL is an enzyme that helps break down triglycerides, which are found in lipid droplets, and you've heard about lipid droplets again from an earlier talk. We also had some evidence that TANGO2 might interact with ACOT enzymes. These are enzymes that regulate the levels of acyl-CoA, fatty acids bound to coenzyme A. Again, you've heard about coenzyme A. So we decided, okay, maybe our hypothesis would be then that TANGO2 is regulating the levels of acyl-coenzyme A within a cell, and that might determine how efficiently fatty acids are going to be metabolized, and the metabolism of fatty acids to generate energy is taking place in the mitochondria. So maybe all this is sort of fitting together in that way. So how are we going to start testing some of this? So we decided to use a new model system, which Chiara Gamberi, our collaborator, helped us to establish in our lab. And that's the Drosophila melanogaster, which is the fruit fly. And we have fruit fly that has a TANGO2 mutation. Well, it actually, not a mutation in the gene, but it reduces the expression level of TANGO2 to very low levels. So essentially the cells are not expressing TANGO2. The flies are not expressing TANGO2. We also have these human fibroblasts that don't have TANGO2 expressed. And we thought, okay, why don't we just, we try to go with the simplest explanations. And so if this has something to do with coenzyme A, let's just put vitamin B5 or pantothenic acid onto the human cells or supplement it into the growth media of the flies, and let's see what happens, right? It was a rather simple notion, and it actually turned out to be an interesting one, in fact. So what we're looking at here are flies and their survival during fasting, which is a trigger of a metabolic crisis in humans. In black, you see the survival curve of wild type flies. Let's just look at where 50% of the flies have died off. It's about 25 or so hours for the controls. For the flies that have the TANGO2 mutation, about 50% of them have died off within about 12 hours. So quite a significant difference between the controls and the mutants. But when we grew those mutant flies in the presence of vitamin B5 supplementation of vitamin B5, that's shown in blue. You can see it's shifted close to where the controls are, suggesting that vitamin B5 was allowing these flies to survive for longer periods of time during starvation. And we also looked at seizures, which is, you wonder how would you see a seizure in a fly? This is a video of when we take a vial. All we do is we raise the temperature to about 42.5 degrees Celsius. And normal flies will not have a seizure at that temperature. And not every fly with a mutation will have a seizure at that temperature. But the TANGO2 mutant flies do have seizures, and they're quite easy to determine to see, as you can see here on this video. And so we looked at the rates or the percentage of the flies that had a seizure as we raised the temperature. The control flies, none of them did. About 50% of the TANGO mutants did. But when we supplemented their food with vitamin B5, that was greatly reduced. And in fact, the time to the first seizure was increased, whereas in the TANGO mutants that were untreated with B5, it took about 20 to 25 seconds to see the first fly having a seizure, whereas on vitamin B5, it took closer to 90 seconds to see that first fly have a seizure. Okay, so vitamin B5 seems to have a beneficial effect on seizures in the flies. Now, flies, intellectual deficit is also a clinical feature seen in most TDD individuals. And so we set up an assay to look at learning, basically. And so flies like to move towards the light. However, what we did is we kind of played a little game with them, and we put a little piece of paper on the light side of this tube that we set up. One side is in the dark, one side is light. On the light side is a piece of paper with quinine, which is bitter. Fruit flies like sweet things, not bitter things. And so we trained the flies over a period of trials of training sessions, and these sessions lasted just seconds. And then we did a test to see, are they learning that the light side, although they like it, also has something bitter which they don't like. And 70% of the control flies learned that, and they stayed away from the light side. Whereas you can see here, the Tango mutant flies, only 30% of them learned that. But when they were given vitamin B5 supplementation in their food, you could see that greatly increased, again, suggesting that vitamin B5 was helping the flies' learning abilities. We also looked at movement disorders in the fly. So the first one I'm showing you here is in adults. And by an adult fly, I'm talking about 10 days. So we can, and we will be aging them longer, but we've done most of our experiments on 10 to 12-day-old flies, which are young adults. So you can see here an assay where we just knock the flies to the bottom of a tube, and we give them a little bit of time, 10 seconds, to climb past what we mark as a finish line. And 80% of the wild type, the control flies, were able to hit that finish line within 10 seconds. And looking at the black line over here for the Tango mutants, again, just maybe 15% of them could pass that line. With vitamin B5, that was greatly increased to near control levels. And two other vitamins that we looked at, we've looked at more vitamins now, including vitamin B9, B12, and I can't remember the other vitamin, but I can look that up if anyone's interested. Not having the same effect as vitamin B5. We also looked at flies earlier in development at the larval stage, and so we can place these flies on a dish, these larvae, I should say, on a dish, and just watch them crawl over a 9-minute period and measure what distance they've crawled. So you can see in black here what the controls, their movement was about 12 centimeters within 9 minutes. The mutants were about half of that. The mutants treated with vitamin B5 were rescued to control levels, and here showing 3 other vitamins that were not having any effect on the movement of the larva. Okay, so these movement disorders are also being rescued. Now, the student who was doing this, Priya Sadi, many of you met her last year at the family conference in Orlando. So she had noticed that whereas the control larva shown on the left seemed to move pretty much left to right on this video, the Tango mutants seem to be moving in a more aimless, uncoordinated fashion. And so we measured that as well. And so the way we decided to measure it was, and I hope I can explain this well, if we're looking at the movement between 2 points, point A and B, if a larva is moving in a perfectly straight line from A to B, then the distance that they've moved, D, is going to equal the distance between A and B, right? So we'll get a value close to 1 if they're moving in a straight line. But if they're taking a more circuitous route to get from point A to point B, then the actual distance that they move to get from A to B is going to be more now. Okay, we actually saw that for the control flies. This is that when we plot D over delta X, we see 1.036 for the controls, and for the mutants, it's much higher. But the mutants that were treated with vitamin B5, that value is starting to come down now. Again, suggesting that this possibly a form of ataxia in flies is also being alleviated by treatment with vitamin B5. And my favorite experiment was this one, where we asked the question, do the flies need to be on vitamin B5 for their entire lifespan in order to see these effects? And again, the entire lifespan of the fly that we're dealing with is 10 to 12 days. So what we asked was, can we give them a little bolus of B5 a day or 2 days or 3 days before we do the experiment? Okay, so what we're showing here are two of our quicker assays. This is the one where we watch for 10 seconds while they climb up that tube, the adult flies. And on the right is looking at seizures. So again, the Tango mutants do not climb very well. About 15% of them are able to hit that finish line. What I've shown you before is when they've been on vitamin B5 their entire lifespan, they've only known vitamin B5 in their food, then that's rescued to near control levels. But even 1 day prior to the experiment, just 1 day treatment of vitamin B5 significantly increased the number of flies that could climb up that tube. And you can see there's a nice steady increase as we move from 2 days to 3 days. So there's a time-dependent increase in their ability to climb. And in terms of seizures, we said there's a lot of flies that are experiencing a seizure as we raise the temperature when they've been on vitamin B5 their entire lifespan. That's greatly reduced. But even 1 day on vitamin B5 significantly reduces the seizure rate of those flies. And I like this experiment because at least for flies, it's never too late to get on vitamin B5 to relieve yourself of the symptoms associated with TANGO2 deficiency disease. And hopefully that translates to humans as well. And I think anecdotally, we've been seeing that it really does. That we've seen from Christina's talk that the reports from parents that as soon as they got their child onto a B vitamin or a multivitamin, that things started to improve, right? So it doesn't seem like it's ever too late, but perhaps there's a better time to start to prevent issues in the long term. Okay, so again, this is the table that I showed you, and with the check marks, these are features that we tested and that were rescued by treatment by vitamin B5. Some of them we're still thinking of ways to look at for the cardiac crises. Some of them are simply not testable. Speech difficulties, right? I mean, if I ever hear a fruit fly talking, well, I got another issue going on there. So we can't really test that. Spells, that's going to be nearly impossible for us to test as well. So we wanted to know if this is only in fruit flies. And so we went to our cells that we have from, we did this on several individuals that have TANGO2 mutations, but I'm just showing you one of them here. There's a complicated slide here, but to simplify it, all the solid lines are control cells, no TANGO2 mutation, and the various colors show you what vitamin they were treated with, or in black, no vitamin at all. All the dashed lines are the TANGO2 mutant cells. Again, the colors are matched to the vitamins that they were treated with before or untreated in black. And what you can see here is all of the dotted lines, the mutants, are clustered down here. This is our trafficking assay, our transport to the Golgi assay that I showed you earlier. So we're just measuring that fluorescent molecule getting to the Golgi. And it's delayed in the TANGO2 mutants, as I showed you, and it's faster in the wild type, as I showed you. And none of the vitamin treatments were rescuing, except for this one dashed line here, which is clustering with all of the controls, and that's the vitamin B5 treated cells. Okay, so again, this is showing that vitamin B5 is also having an effect on human cells, not just on flies. And what I think is really important about this slide is it's showing you that vitamin B5 treatment can functionally replace TANGO2. You don't need TANGO2 as long as you've got vitamin B5, according to this experiment, when we're dealing with a cell. Okay, I'm not talking about a human being at this point. All right, so again, this is what Christina showed you this in the natural history study, and I showed you this part before with the mito cocktail, and you've seen already that multivitamins and B complex are rescuing, are reducing the incidence of a metabolic crisis in these individuals, and so it's very nice that is converging with the natural history study. So we wanted to know what exactly is going on, as coenzyme A is, as vitamin B5 is a precursor to coenzyme A, and as you've heard, coenzyme A can attach to fatty acids, giving us acyl-coenzyme A's, and so we thought there's got to be something happening at the lipid level, and so we did a lipidomic analysis on these fibroblasts from these patients, and this is what's called a volcano plot. The spots that you're seeing to the right of this rightmost vertical line are lipids that are accumulating in individuals that have a TANGO2 that don't have functional TANGO2. Anything in between are not really significant. And this horizontal line is showing us statistical significance. So anything above that line is of interest. And so this can be shown here as a bar graph, just looking at various classes of lipids. And the first thing that struck us, well, two things can strike you. There's a lot of lipids that are accumulating in these cells, and triacylglyceride is really pronounced accumulation in those cells. When we looked at when we starve those cells now, so this is triggering a trigger for a metabolic crisis, more lipids are accumulating in those patient cells, and triacylglycerides are even higher than they were before. They were about 5 or 6-fold. Now they're up to 9-fold or so. What happens when we treat the cells with vitamin B5? So now you can see we've kind of overdone it in a sense. A lot of the lipids are now underrepresented or not accumulating in the patient cells. They're actually at a slightly higher level in the control cells. And a lot of the triacylglycerides, though still accumulating in the cells, are right near the level that we consider to be significant or to be significant or not significant. Okay, so now we're down to about a 3-fold accumulation of triacylglycerides, and this is just easier to see with these two volcano plots. The starved cells and the cells that are treated with vitamin B5. You can appreciate there's a big difference going on here, and vitamin B5 is actually doing something. Okay, and this is just looking at the triacylglycerides just to get rid of all those other spots that you saw. Okay, so we're seeing a change, or triacylglycerides has caught our attention. And so we wanted to know what exactly is going on here. So triacylglycerides, as you heard before, are stored in what's called a lipid droplet. Okay, so this is representing a triacylglyceride, 3 carbons from glycerol, 3 fatty acids represented just by R1, R2, and R3. And when the cells are then starved of nutrients, then what can happen is the triacylglycerides can be metabolized, releasing fatty acids that can then be conjugated to carnitine or coenzyme A and metabolized in the mitochondria for energy. And so what our data is suggesting is perhaps TANGO2 is acting upstream of that. Perhaps TANGO2 first is interacting with a lipid droplet to do something, which we don't quite know yet. And then after that, those fatty acids can then be metabolized. And therefore, in the absence of TANGO2, we're seeing an accumulation of triacylglycerides in the cell. And so on my last data slide over here, I'll show you an experiment that was recently done by Maza Marinfar in the lab, who's working on the lipidomics. And so we take cells, we treat them with oleic acid, which causes production of lipid droplets. There's an increase in lipid droplets. And this is done under nutrient-rich conditions. When we then change the medium and we starve the cells of glucose, we're going to see a decrease in lipid droplets. And so what we want to know is, where is TANGO2 during this process with respect to the lipid droplets? And so Maza looked at that. And so we're looking here at what's called a Mander's overlap coefficient, which is just a measure of the overlap between two signals, the two signals being lipid droplets and TANGO2. And so you see where things start before starvation, the washout of the oleic acid, and that's when starvation was initiated. And you can see within an hour of starvation of the cells, there's an increase in the overlap between TANGO2 and the lipid droplets. And this decreases over time because the lipid droplets eventually disappear within a few hours in the system that we're looking at. So this suggests then that there is a transient interaction between TANGO2 and lipid droplets where triacylglycerides are housed. And so this is giving us, I think I'm at 2-minute warning here. So I'm going to go to my 2-minute offense here and try and finish up with my conclusion. So basically, we've got vitamin B5, which seems to be rescuing these the phenotypes that we're seeing in the TANGO2 flies and also in the human fibroblasts that we obtained from TANGO2 patients. We're not sure what the mechanism is, but something is happening at the lipid level. And particularly catching our attention is the triacylglycerides that are accumulating in those cells. That imbalance seems to be affected greatly by treatment with vitamin B5. And I've shown you that TANGO2 is transiently associating with lipid droplets. And it's those lipid droplets that do contain these triacylglycerides. And so what we're doing is taking some biased approaches right now. And because of some of the similarities in phenotypes between TANGO2 individuals and ATGL deficiency disease, we thought maybe TANGO2 is affecting lipolysis. So ATGL is the enzyme that will start breaking down triacylglycerides. And so we want to know if TANGO2 is affecting the ability of ATGL to hydrolyze triacylglycerides to release those free fatty acids. So we're taking some biased approaches, setting up some enzyme assays with ATGL and TANGO2, and as well as with ACOT, as I briefly talked about before with TANGO2. And we're going to continue the work with the flies, looking at organ-specific drivers. So we'll be able to get rid of TANGO2 specifically from brain or specifically from heart, and then ask if vitamin B5 or any other combinations of vitamins are beneficial to those flies. And that's the power, I think, of the fly system, is we'll be able to test some of these things in terms of even antioxidants, I think was a question. Some of these vitamins are antioxidants. And so what if we put in all of the B complex vitamins, where B2, I believe, is an antioxidant? Are we going to see a better effect there? Okay, so that's sort of where we're heading with the work. And I just want to thank, Priya Sadi, as I mentioned, many of you met her last year at the conference, did a fantastic job on all the fly work. That's being continued now by a new student in the lab, Rozmir Shokoi. And Miroslav Milev did the mammalian work, and now continuing and doing a great job with the lipidomics work and these enzyme assays is a PhD candidate in the lab, Maza Marinfar. And Alia Naz is another student in the lab who's also working on TANGO2 in various cell systems and various different types of cells, including yeast cells and other types of mammalian cells. So thank you for your attention. I'll be happy to answer any questions.